Well, good. Yeah. Okay. And I assume that the people on Zoom can hear me. Otherwise, put it in the chat, and then the technical side will work. Yes. Okay, it's a pleasure to be here in Istanbul. Again, that's my third time in Istanbul. I think 6th time in Turkey. Never for vacation, always for work. And this picture, I took this picture 11 years ago. You, of course, recognize where it is. And 11 years ago, we gave the first ever HADDOCK workshop in Istanbul at BioExcel University. This was on the terrace of the university, looking over the Bosphorus. It was beautiful. It was a great workshop, and it was my second time actually in Istanbul. I visited in 2008 the first time, also to give a talk at the same place. So this is the program for today, at least for my lecture part, and then we'll do some hands-on session on Truba resources. So I'm going to give you a general introduction. Then I want to introduce integrative modeling, the way that we are doing that using HADDOCK. And I will introduce you to a new version of HADDOCK, which you might not yet be familiar with. Some of you might have used HADDOCK in the past. So this is HADDOCK 3. That's the one that we're going to use also in a hands-on session to do some real calculations. I'm going to very shortly touch on antibody-antigen modeling, because that's also something that we're going to do in the practical part. And then I want to show 2 short topics about modeling different types of complexes, protein cyclic peptides that have their own challenges, and then at the end, if time allows, protein glycans. And I will finish with some general conclusions. So why study protein, the social network of proteins? This is the center of Utrecht. So here in Istanbul, you have the Bosphorus, which is a nice piece of water. In Utrecht, we have a canal in the middle of the center. It's actually the old Rhine going through the center. And you know, maybe some of you know Amsterdam. Amsterdam is very famous, of course. But one thing Amsterdam does not have, which makes Utrecht much better, are those terraces at the water level. Okay, these used to be places where they were storing goods. They were bringing goods by boats and then storing in the cellars. And now all these cellars have been converted into restaurants. And it means that there is a lot of social interaction at the macromolecular level on those terraces. But everything in life is, of course, governed by interaction at the microscopic level between biomolecules in our cells and communication between cells as well. So if we want to understand how things work, we need to understand interactions between biomolecules. And this brings me to the concept of the interactome. So we have the genome, the genetic information. That's basically the hard drive of your computer. It stores information, but on its own, it cannot do much. The proteins are really the CPU of life. They are doing the work. And they are doing the work by interacting. So you have the genome, then the proteome, which is all the expressed protein in the cell. And the next level of organization is the interactome, which is the map of all interactions taking place in a cell. And this is a picture of such an interactome obtained through some high throughput experimental methods. There is a lot of limitation with those methods, so that's only a fraction of the problem. And there's also a lot of false positive data in this. But what you see here is a lot of dots. The dots will represent the proteins, the players, and a lot of connections. And the connection indicates that 2 proteins are interacting in one way or another. And if you start doing statistics on those, you will realize there are many more connections than there are dots, meaning that proteins during their lifetime will interact with many other, the average will be 8 to 10 other biomolecules. This network is not going to be static. It's going to be rewired as function of when you are in the cell cycle, as function of post-translational modification, as function of localization in a cell. So it's a dynamic network of interaction, basically. And as in real life, you know, miscommunication between humans often give rise to problems. Miscommunication at the cellular level is also at the origin of disease very often. So if we want to understand how the network works, we need to add the structural dimension to it, to basically look at the complexes. And that brings me to the structural biology of interactions. And in this context, historically, we have three methods that allow you to study biomolecule at atomistic resolution. That's X-ray crystallography, the oldest of all, nuclear magnetic resonance, which works in solution or solid state, and very importantly for complex these days, cryo-electron microscopy. So those three techniques will give you coordinates of atoms in space for large biomolecules. But next to those, there is a lot of other experimental methods, and this is by no means a list of everything that's out there. But you can use mass spectrometry, for example, to study interactions. This will give you some bits of information, small angle scattering. This will give you shape information. Electron paramagnetic resonance, for example, will allow you to measure some distances. You can do biochemistry, mutations, running a gel, seeing if the interaction takes place or not. And all those techniques will give you pieces of a puzzle that you can only solve if you combine those pieces with computational methods. Which brings me now to the left side of the slide. So what you see here are the classical modeling techniques that have been used since a long time to basically generate models of proteins, of their complexes. So you can try to use homology modeling by analogy to something that should already have been solved. You can try to use molecular dynamics, but to predict complexes, that's very expensive and it takes a lot of time. And the method of choice over the years to basically model interaction has been docking. But of course, these days we are living in an AI era. Since 2021 with AlphaFold 2, Nobel Prize last year in chemistry for structure prediction. So since the early days of AlphaFold, people realized that you can also use it to predict complexes. And then our DeepMind came with a version specific for complexes, AlphaFold Multimer. A lot of work has been going on, and a lot of different methods have been published where you can look at complexes. We have balls, we have Chi, we have OpenFold. So this is a field which is moving extremely fast. And more recently, last summer, basically AlphaFold 3 was released. Prior to AlphaFold 3, the predictions were limited to protein-protein interactions or protein peptides. And with AlphaFold 3, you can also do protein nucleic acids and even some small molecule modeling in there. All those methods rely heavily on multiple sequence alignment and the concept of coevolution, meaning that they will have their limitations for systems where coevolution is not playing an important role. And one of such systems are antibodies and nanobodies. So there is still plenty of need and space for the more classical methods. So those methods here will be typically what I would call physics-based or maybe statistics-based, based on analysis of non-structure, while the AI methods are basically relying a lot on sequence alignment, but then on the training on the large data set of known structures. But for today, we will concentrate on, at least for this morning, we'll concentrate on docking. So molecular docking in a nutshell. So given the structure of two proteins, so most docking methods, basically you need a starting point for the modeling. So we assume that we either have the structure of the component of the complex, or we can generate those. And these days, AlphaFold and such method will allow you to do that quite easily. And then the problem is, so given two structure, you can say, well, I'm going to fix, if you want to do a systematic search of all possible solutions that you can generate where the molecules are interacting, you can fix one molecule in space. And for the second molecule, you have to sample all possible rotation of that molecule. And for each rotation that you sample, you have to sample all possible translation around the first one. So there you will be doing a systematic search of all possible interactions. And for each of those models that you generate in that way, then you need to calculate some kind of score that's going to tell you if this is a good information or a good model or a bad model. And in these scoring methods, what has been used in the initial days, so docking was invented, say, in the late 70s, the first methods, it was purely shape complementarity. So all complementarity are the surface of the protein. And then later on, people added, physics, chemistry, electrostatic, van der Waals interactions, and then you can also add many other methods. But this is basically docking in a nutshell. So what we have to do when we do this kind of computation, so we have to sample some kind of interaction energy landscape. We don't know what the landscape looks like. So you might say, I'm going to do this systematic sampling, all possible translation rotation. Or if you have some knowledge, you might do a more targeted sampling. But basically, sampling is basically generation of models. And then we have to score those models with some kind of function, which can be the energy. So in this plot, you have here the interaction energy, but this doesn't have to be an energy in principle, with the hope that your function, if perfect, will allow you to identify the solution to the problem that corresponds to the, say, native state of this interaction. Now, if you have data, and this can be experimental data or predicted bioinformatic data, you might choose to use the data to bias the sampling, and this is where integrative modeling comes from, or you might use the data to bias the scoring. You want to select the models based on their fit to the data that you have at hand. And these are different strategies. So you have software, docking software out there that are doing a global search. Example of that will be Prof, for example, HADDOCK. And you have software where you basically direct the search to a small region of space defined by the data. And this is what HADDOCK has been developed for. So the advantage here is that you can focus your search and spend more time in the region of space which should be relevant for the interaction. The danger is that if you have bad data, wrong data, you're going to search in a region of space and you will never going to get the right information. Okay, so you have to trust your data and you have to have a way also of dealing with false positive information possibly in your data. Here, you're going to sample much more of the entire interaction space at some resolution, because this is an expensive sampling in principle. So hopefully, in that case, the right solution is somewhere in the ensemble that you generate. So when we speak of integrative modeling in the field, we are speaking about combining different sources of information together with modeling techniques to basically predict the structure of a complex. And you see here some of the sources I already mentioned. So you can do mutagenesis with some biochemical assay or biophysical assay to measure binding. If you find a mutation that destroys your complex, you would assume that this is important and most likely it's at the interface of the complex. You can try to predict binding sites. You can try to predict even contacts. That will be the concept of coevolution these days. You can do NMR to define binding sites. NMR is very good for that. You can, from NMR, also get orientational information. Cross-linking, add small molecule that would attach covalently to your complexes, and you detect those typically by mass spectrometry. That's very powerful these days, and you need very little samples. So this will give you some distance information now. Not very accurate because you don't measure the exact distance. You just only know that two residues are connected by a flexible linker, and the linker could be doing that or it could be doing that. So you don't know what the distance is. HD exchange, also very powerful together with mass spectrometry. So protection from exchange will indicate possibly an interface. And all this information can be then used to either score or drive your modeling process. And this is what we have been doing now since more than 20 years in HADDOCK as integrative modeling platform. So HADDOCK supports all these different types of information, basically anything that you can transform into this residue is important or this is some kind of distance can be used into HADDOCK very easily. We can also use shape information. I started explaining docking with 2 molecules, but of course, many complexes consist of more than 2 molecules. So you also need to be able to do this kind of modeling. If you do the systematic sampling that I explained, translation, rotation, all the docking methods that do that can only handle 2 molecules. Because once you add a second molecule, then the sampling that you have to do is exploding. So there are not so many methods that can handle more than 2 molecules at the same time. So HADDOCK being one of those. Symmetry is in principle also information, so you can restrain the symmetry of the system. So what kind of molecule types can we currently handle? So it started with proteins, but so protein peptides. So peptides are no different than proteins, but peptides have more of a flexibility challenge. So you need to play some tricks with those. Nucleic acids, we support both DNA and RNA. We don't have any support for modified bases like methylation. That's something we are currently started working on. Glycans, Glycosylated proteins, so protein carrying a glycan. So if you have glycosylation, you can include it in your system. You can do small molecule docking with HADDOCK. You don't want to do virtual screening campaigns with HADDOCK. That will not be very efficient. Better use the small molecule docking software for that. But if you have some experimental data on your system, then HADDOCK is a good choice. Shape, so we can define arbitrary shapes. And you see two examples here. So this could be a shape coming out of a Sachs experiment, for example. Small angle scattering, you do a shape reconstruction. And then we have a way of defining restraints where you say basically your molecule should fit into this shape, should overlap with the shape. And this will be another representation of some biological system. What could that be? Two planes. What could that represent? A membrane, yes. So that will be a very coarse representation of a membrane. And there you could say that, okay, if I have a membrane protein binding to some, or two membrane protein, you could say, okay, those membrane proteins should basically sit between those two planes, okay, and not sample the other parts. Or you might have a membrane protein binding to something else. So it's basically describing the membrane without having to explicitly describe the lipids. Now, the type of restraints, I mentioned already quite a bit about that. So any kind of distance, we distinguish between ambiguous and unambiguous distance. Unambiguous will be a distance between 2 points. That's the classical distance that you think of. But the concept of HADDOCK, and it's going to be in the next slide, if I'm correct, is to be able to use what we call ambiguous distances, which will be distances between many points. Basically, you don't know what the exact partner is, but you have multiple possibilities. So this defines a network of distances rather than a single distance. Okay, so how do we encode this? How do we define this ambiguous restraint? So often the information that you might have is that these residues are important for the binding. And if you are lucky, you have this information on both sides for both proteins. But what you don't know if you have this kind of information is what is the pairing which is done. Because you know this residue is important, so most likely it's making a contact, but I don't know with what, okay? And in a HADDOCK concept, we use this information to define ambiguous restraints by defining a network of distance from this residue to all the residues that are in the interface or predicted to be in the interface on the other side, okay? And we calculate all possible individual distances. We sum those as 1 over the distance to the 6th power. So 1 over r to the 6th. It's dipole-dipole interactions if you are an NMR person, but also the attractive part of a van der Waals potential if you are more a molecular dynamics type of person. So you might have 1,000 distances that you calculate for each amino acid here at the interface. So you calculate all pairwise distances to all amino acids defined as being potentially at the interface on the other side, sum them as 1 over 2r6. So as you add distances to this sum, the sum becomes larger and larger and larger. And then you take the inverse 6th root of that sum, and that gives you a single distance, okay? And that distance will be shorter than any distance that entered the sum. That's simply mathematics. And that's the distance, so that's the distance that we basically calculate when we define a restraint. We say this residue should be at the interface, so we define one distance restraint between this residue and all the residue at the interface on the other side. We calculate that distance from all the combinations. If that distance is within a cutoff we defined, we say the restraint is satisfied and the model will satisfy the information. And we use a potential function which is harmonic for a small part and then becomes linear, so that if you don't satisfy the distance, the energy will still go up, but the force becomes constant. The force is the derivative of the energy minus the derivative of the energy, which is related to the slope of this function. So the slope in this section, in this region here, will be constant. So meaning if you don't satisfy the distances, the force don't become crazy. Your simulation are still stable. You know, if you have very large forces and you do molecular dynamics, this is when you get trouble and your simulation explodes, basically. So that makes that we can also deal with false positive information without blowing up the system in our calculation. So that's the secret behind the ambiguous restraints. What we also do by default in HADDOCK is to basically randomly delete 50% of the information that we put in. And that's a way of dealing with false positive information. course, if you can turn off this random removal, by default, it's on. And it means that if you have bad data that you are using, from time to time, you're going to remove them and hopefully generate good models. But from time to time, you're also going to remove the good data and generate bad models. And then it will be up to the scoring function that you are using to distinguish, hopefully, between those. So how do we do the docking protocol itself? So we don't do this systematic sampling of orientation. We use a combination of energy minimization and molecular dynamic simulations. And we repeat this process from different starting conformation of the molecule. So we separate the molecule in space. We randomly rotate the molecule in space. And from those positions, we start an energy minimization first, based on the data that we put in that will draw the molecule together. And we optimize then at a later stage the interface by starting to do very short molecular dynamic simulations. So this is how this minimization basically proceeds. So this is rigid body minimization. So only variables that are changing or the degrees of freedom, as we call them, will be translation rotations. The molecule at this stage are rock solid. This we might do 10, 100,000 times. That's the kind of sampling that we are doing by default. After that, we do a first selection of models, so what we'll be scoring, and we take a few hundred solutions, and then we go into a slightly more expensive refinement stage. So now, the rigid body minimization, we're speaking of seconds, and now we're speaking of tens of seconds to minutes for this refinement process. And here we refine the interface of the molecule. Okay, so the interface becomes flexible. And if you look at this movie, you see that the side chain will start to move here. That's what's happening there. And later on, the backbone also becomes flexible. Look at this loop here. It's going to flip over in a bit. It just did. So in that way, you can model small binding-induced conformational changes. So this process is actually torsion angle molecular dynamic simulation in torsion angle space. So the variables are not position of atoms. The variables that we are modifying in those are rotation of angles. And this allows you to freeze some rotation very easily. So we basically only release for the dynamic the angles of the residue that are at the interface of the molecule. And the molecule themselves, they can move freely in space. Okay, it's different than doing position restraints in molecular dynamics where things are frozen in space and cannot move anymore. So that's the advantage of the torsion angle. We used to put then this system into water, a layer of water, no periodic boundary conditions if you are an MD person, and run a very short simulation. And by short, I mean here a few tenths of picoseconds, so it's peanuts. This is basically only optimizing a bit the energetics. These days, we don't do that by default anymore. We just minimize the system because it doesn't change the conformation anymore. So these are the three stages of the, say, classical HADDOCK, so rigid body minimization, simulated annealing, and refinement in explicit solvent, or simple minimization these days. And these are the scoring functions that we use to select or rank the model. This is not what is used for the calculation. The calculation uses a full force field. We describe bond angles, diedrals, impropers, electrostatic, van der Waals, the restraints. But for the scoring, we have this very simple linear model. So these days you will call, this is also artificial intelligence. It's a machine learning model. It sounds much fancier. But this was derived 20 years ago, more than 20 years ago. It's a very simple function. It has very round number. So you see, this is the final term here. So in the previous term, so you see this is buried surface area between the molecule, the amount of surface buried between the molecule. The green one, an energetic component, and that's the dissolution term. So we have 20% of the electrostatics. It is the intermolecular electrostatic energy, full van der Waals energy between the molecule. Dissolution that measure basically the bonus that you get by removing water from the interface or the price that you have to pay. If you desolvate an hydrophobic surface, usually you gain energy because water and oil do not like to mix. But if you desolvate a charge, you usually have to pay a price because it costs energy to remove water from a charge. So this is what this term describes. And the last term describes basically the experimental data that we put in. So 20 years ago, this was optimized on 5 complexes and chemical intuition. We have been revisiting that. You know, these days, students want to do machine learning, so we give them a lot of data, and you say, okay, go optimize the scoring function of HADDOCK and try to do a better job. And then they come back to me, and instead of 1.0 for the van der Waals, they say, I have a much better function. It's 0.975532. Say, that's nice, but the function is not robust, is not accurate enough that we can justify using all those digits. So I like to have these round numbers. They make sense to me from a chemical intuition. And they are robust also. That's also important because we use this function to score protein nucleic acid interface and to score protein-protein interface as well. Okay, it's not perfect. It's physics-based, but it still does the job. We have also been doing machine learning, deep learning to try to optimize a network to predict the quality of the model. So this is one such example. So it's the deep rank software, which is basically transforming a 3D representation of a complex into a 2D graph. And then we train the network to basically predict the quality of the structure, quality of the model. And some of the features that are used originally was the position-specific scoring matrix. So this is something that you calculate from multiple sequence alignment. And it takes a bit of time to calculate those. You need all the sequences. And more recently, we have been replacing those features by features coming out of a language model. So this large language model. So this is ESM2, which was trained by Meta. So you don't need multiple sequence anymore. It's much faster. And next to that, you have amino acid type, charge, polarity, these kind of things. So this is going to be integrated also as a possible scoring function for in HADDOCK 3, actually. That's one of the modules. But currently, it's not in a standard version of HADDOCK. Still, it's not like it's, in some cases, it's slightly better than the HADDOCK score. In many cases, it's about on par to the HADDOCK score. So, it's still hard to beat the good old physics, I would say. So most of our users are using HADDOCK through a web portal. And you see a snapshot here, which is accessible from WNMR Science UNL. So we have been operating this server since 2008. And we can do that because we are running on EOSC high throughput computing resources. And in the presentation before, I heard also that Turkey through Truba is part of the EGI Federation. I think you have cloud resources. We are not yet using those, but I'm not sure where the person was presenting, but I surely hope that in the future we can make use of those. I know that we have a service level agreement where you committed some storage, which we are not using, so I will probably knock on the door at some point. But it means that the computations that HADDOCK is doing in most cases are being distributed to a network of computers across Europe, but also goes to Asia a little bit. We cross the Atlantic from time to time. And this is what is allowing us to serve basically the community. Now, this is the current community. This is, I think it's an old slide, this one, because now we are reaching, I think, 59, and we have 161 countries covered. So this is our world domination card map. This is the user growth. So we're still in an exponential phase of growth. Even with AlphaFold and all the AI things happening, we have not seen yet a slowing down in terms of usage, which is good for us. Monthly registration. The last 2 months, last couple of months, it's almost double. So we had for last year about 1,000 per month new users registering, and now the last 2 months we are reaching 2,000. So not sure what happened there, but it's going up. This is the number of submissions that the server is processing per month again. So around 10,000 per month, and now we have peaked up to about almost 15,000. These are docking runs, and each run that a user submits will translate in a few, several hundred jobs that are sent to this distributed resource across Europe, and in the future, Turkey, hopefully. Okay, and this is a distribution of users over the different continents. And actually, this is the number of users that have registered in a server in Turkey since the early days. So it doesn't mean that 1,000 users are active today, but since 2008, this is the number that we have been accumulating. Okay, to support, so when you develop software, if you want your software to be used, you have to provide support. That's very important. You have to provide tutorials. You have to give such workshops. And we have a support forum online, which is operated through BioExcel, the Center of Excellence for Computational Biomolecular Research. There you can also find support forum for the other software that you're going to hear about in this workshop. So if you have questions about HADDOCK, that's where you can go. It's freely searchable. If you want to post question, you have to register. And you can also try to answer questions. So it will be nice if the community also grows at some point that users start to answering questions. Now, HADDOCK is not the only tool that we have been developing over the years in my lab. So we have other tools. Most of those are also provided as web services. And all of those have in common that in some way or another, they have to do with analyzing or predicting biomolecular interactions, okay? We're going to use at the command line, for example, PDB tools today in the practical. Okay, so this was about HADDOCK and 20 years of development and just the classical way. But in the context of the BioExcel project, which I already mentioned, we have been working since several years on the next generation of HADDOCK, which we call HADDOCK 3. And you see HADDOCK 3 now is a collection of puzzle pieces which are kind of disconnected from each other. So this is the static workflow that I show you. So on the server, you can change a lot of parameters. There's probably 500 parameters that you can play with if you know what you are doing. These parameters will change some of the behavior of HADDOCK, but you still have to go through those 3 stages. So even if you want to say, I don't want to do rigid body docking, you're still going to go through that module, write files to disk, which have done nothing, basically. These are the same as your input files. And so it's a static workflow in which you can tune parameters. So in HADDOCK 3, what we decided is to basically break the code into modules. So we have now a catalog of modules. And now you can combine these modules to do different things. So you can define workflows. And if you say, I'm only interested in using HADDOCK for refinement purposes, I want to do the water refinement at the end to maybe remove clashes from a model I generated with AlphaFold, you can do that without having to go for the other modules. So you have some flexibility in your definition of workflow. This is the current list. I think it has even been extended. I see some pull requests recently. So we have, so one thing which is required for everything currently is topology. So you need to build the topology of your system. This will also build all the missing atoms in your system. You know, if your side chain is not complete because you are using a crystal structure where the side chain was disordered, HADDOCK will build for you those missing atoms. We have a sampling set of modules. So the rigid body that you see here is the classical one in the HADDOCK 2 series workflow. So the first step of HADDOCK workflow. And we have 2 other modules that are third-party software. So LightDock is another integrative modeling software which can model binary components, partially developed also in Utrecht. And GDock is a docking software based on a genetic algorithm developed by one of my group members. We then have the refinement module. So this is the second and third stage of the classical HADDOCK workflow. So the semi-flexible refinement, which I described, final energy minimization, possibly short molecular dynamics in water if you want. And now we added also a third-party module which basically uses OpenMM, and you can run molecular dynamic simulations within a workflow. You probably don't want to do that if you're going to run microsecond simulations. So if you're going to go for microsecond simulation, use Chromax or use OpenMM, but directly and not in the context of a workflow. But if you want to add some sampling, it can be now part of a HADDOCK workflow. We have scoring models. So these will be used if you have a set of complexes, not per se generated by HADDOCK, but you want to score those. You can use those scoring modules. Some do only minimization, so building the missing atom, minimizing, scoring the interfaces. And the other one does a short simulation in water as well. And we have a number of analysis modules that are here. So some of those have to do with, so we, in the HADDOCK workflow, at the end of the workflow, we cluster the solution that we get. So we put basically similar solution in bags so that you don't have to look at single solutions, but you look at clusters. So that diminishes the number of possible solutions, and we do a cluster-based scoring. So there are different ways of clustering by contact or by RMSD. We can calculate different types of RMSDs. We have selection modules where we say, we do rigid body and then you want to select the top 200 models to go into the more expensive refinements. So you will have to put those in between. We have analysis modules, so you can generate a contact map of your system at the end to visualize the contact. And we have added an alanine scanning modules where you will have, for complex, it will automatically by default detect all the interface residues and mutate each residue in turn to an alanine and give you a difference in score, basically. Then you generate a plot. This is useful, for example, to analyze the energetic of your interface, which residue are important and things like that, or maybe possibly design mutations. You can also mutate to any other residue that you wish. So. And deep rank is in a process of being added. And this is how a workflow looks like. So that's a TOML format for the more bioinformatic computer people among you. It's a simple text file, basically, with a collection of modules. The one that you see in red here will be basically mimicking what the HADDOCK server does. Okay, so we build the topology. We then do a rigid body minimization. We select the 200 best model. After that, we do flexible refinement, final minimization. Capri-eval is just generating some statistics. It's nice to get plots and things like that. It's using the best model as a reference in that case. And then we do clustering, selection of clusters, and again, an evaluation step to get the cluster statistics out of it. And you only need to specify parameters that you want to modify. So all the default parameters you don't need to touch in principle. Of course, you need to provide also restraints. So in this case, you see here a list of, these are distance restraints file, ambiguous distance restraints file. So we're going to see those also in the hands-on. Now, if you use the server, the result page of the server will give you an analysis of your cluster, give you the cluster statistics and some graphical analysis. If you are using HADDOCK 2 version on the command line, you will have to do all of that by yourself. Okay, you will have to do this post-processing analysis manually. In HADDOCK 3, those reports are automatically generated. So there is a report, basically an HTML file, which will give you the table with the different clusters, the statistic of each cluster, plots that show you different energy score with respect to RMSD or fraction of contact, and you can also visualize the cluster. So this is basically, from the command line, you can basically visualize those things. And we have been working with the Dutch eScience Center on the interface for HADDOCK 3, a graphical interface, basically. So we have the HADDOCK 3 web app, which is available from this repository. In principle, you should be able to build it quite easily. It uses Docker containers for that, and you could operate this interface from your laptop directly or on your own cluster. And this will form the basis for a HADDOCK 3 web service that we are currently developing in the context of another project. So in the future, we will hopefully have a HADDOCK 3 server as well, although the compute model there will be slightly different. Okay, so let's move to the first very short example here. I just want to touch on antibodies because we're going to do a practical using antibodies. And in antibodies, even if you don't do any experiments, you already have information because we know that in antibodies, the regions that are involved in the binding are the tips of the light and heavy domain. So antibody consists of two heavy chains, the more, say, gray-brownish chains here, and the light chains, which will be more the purple chain. So they're in principle two binding sites. One region is here and one region is here. And there are 3 loops from the light chains and 3 loops from the heavy chains that are contributing to the binding. And those loops are heavily changing, mutating. So there's a variability in the length of the loop, especially the H3 loop, so the third loop from the heavy domain. And they are heavily changing in the identity of amino acid. And it's part of the way that antibodies are produced, so that there's a very high variability to adapt to new threats, new viruses, new bacteria. So by definition, they have to evolve very quickly to be able to respond to threats, which means that there is no conservation signals in those regions, okay? So this is the reason why AlphaFold and related methods have a bit of more hard time to predict antibody-antigen interaction. So anyway, so for antibody, even without any knowledge, we know the binding site is in this region. So that's information that we can use in HADDOCK. In this paper, we benchmark, so this was 2020, different software that can use information in different ways. So we tried ClusPro, ZDock. These are more classical docking software sampling the entire space, and they can use data to filter solutions. And we benchmark HADDOCK and LightDock. So those two software are more integrative software in a sense that they can use the data to bias the sampling as well. And here we show that actually using the data directly to drive the modeling process, meaning HADDOCK in that case, gives you better results. But we also show that if you don't have any information on the binding site, on the antigen site, it's still a very hard problem to solve. Okay, the success rates there are pretty low. After that, we have been working more on basically now doing this kind of modeling from sequence only. This is a bioinformatics paper published last year, October, where we basically use AlphaFold and Immune Builder and different other AI tools to generate models of antibodies, but also of the antigen, assess how good they are, and then use those to do antibody-antigen docking with some knowledge about the binding site on the antigen site. And we compared that also to the performance of AlphaFold. And recently, so this is out since on bioRxiv very recently, so we use AlphaFlow, which is another AI model which has been developed to basically sample conformations a bit better. And this is very useful in the context of the H3 loop of antibodies, which is challenging to model because of its flexibility and length. So if you want to read that story, you can go there. And very soon on a server near to you, this story is going to pop out, which is basically looking at nanobodies, which are like a mini version of antibodies. And nanobodies are even probably more challenging in the sense that the H3 loop, they also have this binding loop, which is very important. It's even longer. So here, conformational sampling is even more of a problem. So just to give you the conclusions, we get our hands dirty on antibodies in the practical. So the current tools are quite accurate in generating conformation of antibodies. So we can use AlphaFold, Immune Builder. Immune Builder is very fast, so that's actually quite a nice tool for that. And in those papers, we show that we can use HADDOCK in a very limited sampling scenario, provided you have some data on the binding site on the antigen. That's what we're going to do also in the practical. And the model diversity is here the key to the success rate. So sampling conformation prior to docking to increase the diversity is the way to go. But there are still scoring issues. So scoring function is not perfect, but also AlphaFold is struggling with identifying right solutions. So let's move into another topic, protein cyclic peptides. So this is the work of Vicky, former master student in the group, and Siri, former postdoc. And here we have been looking at modeling cyclic peptides. So cyclic peptides are interesting because as therapeutic, they have enhanced metabolic stability. The cyclization basically stabilizes the peptide and makes it less susceptible to proteolysis. The classical example of cyclic peptide, which used as a drug, is cyclosporin A, where you see the structure here. This is a molecule which has been used probably in the 80s, also when the first enhanced sampling molecular dynamics methods were developed, and this was before metadynamics was even, the name was invented. So there was another type of molecular dynamics at the time. I don't remember what it was called, but it was in a group of Wilfred van Gunsten. So the problem with cyclosporin is if you do NMR of cyclosporin in water, you will find one conformation. When it binds to its target, basically, it flips its conformation inside out. Okay, and if you have a cyclic structure, it's not so simple. Think of building a 3D model with a ball and stick of a cyclic peptide, and then you want to flip it inside out. That's quite a conformational changes that you have to make. So when the peptide binds to its target, it flips inside out. And that's a huge conformational changes. And this is what has been used as a toy model, basically, to test enhanced sampling molecular dynamic methods. And there are very few studies so far that have been looking at those. So now we also see that AI methods start to play with cyclic peptide as well by doing basically extending the sequence of your peptide and make it introducing a cyclic component into it. So we put together a benchmark data set where we know the structure of the complex, basically. That's what you need if you want to optimize, develop new protocol, new methods. You need to be able to benchmark things. So we have 18 cyclic peptides in which the cyclization is through a peptide bond. So it's the backbone which is cyclized, okay? And we have 12 peptides where the cyclization is through disulfide bridges. So it means that the termini are still free, but it's disulfide that are basically closing. And in terms of length, you see that they range between 6 amino acids, so very short one, up to about 14 amino acids. And of course, the longer the peptide, the more the conformational challenges will be in terms of modeling. So these are examples. So this is a short one, short cyclic peptide. So this will be a long one, 14 residues. Cyclization is through the backbone, but there is also a disulfide bridge. And this is, again, a long one, 13 amino acids, where the termini are free, but there is a cyclic bond. The cyclization is for a disulfide bridge. So the first step is to generate, so if you're going to model a new complex, of course, you might not have the structure of your cyclic peptide. So we need to generate cyclic peptide conformations prior to the docking. And the way we do that is first to build the peptide either in an extended conformation, polyproline 2, or beta sheet. You see that there are some turns in those peptides that are drawn here. Proline induce kinks in the structure. So if you have proline amino acid, you expect kinks. But basically, we just build those peptides into extended conformation. And then in this protocol, what we are doing is to define distance restraints between the termini or between the sulfur atom in the case of a disulfide bridge. And we use HADDOCK as a refinement module. So it's not docking, it's a single structure calculation. So it's like an NMR calculation, basically. Actually, the initial protocol in HADDOCK were derived from NMR structure calculation protocols. And this allows us to bring the termini together, okay? Once the termini are close in space, then we can establish the bond, and then we have the proper chemistry, and then we refine the system, okay? So here, we start from 2 conformation, polyproline beta sheet, we cyclize, generate 10 structure, and then we repeat this optimization process many times, do clustering, and select an ensemble of 10, of 30 or 50 conformation out of that. One of the secrets to get, so what we want to get is not structure of the peptide in solution free, but what we want to get is mainly conformation that will resemble the conformation that you expect in a complex, which might be this inside-out conformation if you think, if you remember the cyclosporine story. So the way to get more bond-like conformation is to turn off electrostatics, actually. So no electrostatic interactions. We only calculate with van der Waals. Because if you turn on electrostatic, what you end up with is that a lot of the polar and charged amino acid want to be in the center of your peptide and make interactions because this is very favorable. And what you get on the outside is more hydrophobic. And when you bind to your protein, often it's more the polar residues that are going to make interactions, and you will need to flip it inside out. This flipping inside out is not going to happen during the docking. That's too much of a conformational change to ask. So this meant at the time running HADDOCK 2 series multiple times, so one time here, a couple of times here, doing clustering, doing a lot of manual analysis. In HADDOCK 3, this is a single workflow. So you can do the entire thing starting from the sequence, from the starting conformation all the way to first cyclization, then refinement with the bond, then clustering, and then selection of model. So that's one advantage. And one thing you could not do in the previous version, we can do now in HADDOCK 3. And there is an example for that in the example directory of the software if you install it. Okay. So we do this cyclization protocol, we sample multiple conformations, and then we give an ensemble of conformation to HADDOCK. So in this case, the protocol takes 50 starting conformation of the peptide, and we don't do 50 docking runs separately. No, we do one docking run with 50 starting conformation, and we use those internally. So our sampling in this case is 5,000 rigid body model and then 400 for refinement. 5,000 here means that we are writing to this 5,000, but the effective sampling that we are doing is in the order of 50,000, because internally we sample many times before writing something out. We add more flexibility to the system during the refinement, so we have a longer protocol to give more time to the peptide to adapt its conformation to the binding site of the protein, basically. And this is the result if you do the benchmarking. So what you see here is the quality of the model expressed as function of the fraction of native contact. So this is the fraction of contact which is consistent with what you see in the crystal structure in your reference structure. Okay, so if all the residue contact are reproduced, you will get 100%. And in a field for protein peptides, we defined if you have 20% of the contact consistent, this will be an acceptable model. It's not great, but it's acceptable. This is based on Capri statistic, and Capri is the critical assessment of predicted interaction. That's an experiment for blind prediction of complexes. You might have heard of CASP. CASP is for structure prediction, although in CASP, there's a lot of complexes now entering the field as well, and CASP run every 2 years. The last CASP prediction season was last summer, and there was a meeting in December to assess the results. Capri runs whenever there is a target coming our way. So we are not bound to a given time of the year, but it runs at any time in principle. So 50% will be a medium quality solution, and 80% will be a high quality prediction. Now, what you see on the top row is if you start from the hollow receptor structure, so no conformational change on the protein side. This is just for benchmarking purposes because you will never have this solution, this case typically. The bottom row is the more interesting one because here we start from the apo-receptor structure, so the free form of the protein, and the peptide model by this cyclization protocol. Now, what do you see in all those plots? On the x-axis, this is the number of models that you consider after scoring, after your ranking. So 1 means if I only look at the top ranked model, and the y-axis gives you the success rate, basically, which fraction of the complexes is acceptable or better. So you see in this case, more than 80% of the benchmark, we get an acceptable solution on the top model. It's not great. Most of it is this light blue, but we have a few cases with high quality. If you look at the top 10 or even the top 5 solution already, that you see that within the top 5, 100% of the case, there is an acceptable solution. So that's quite nice. And this is no clustering yet here. So this is for the backbone data set. So this is cyclization through the peptide bond. And on the right side, you have the same, but then for the disulfide data set. So this was, I was still here. Okay, so you see here that the disulfide, there is more freedom in the peptides because the ends are not cyclized. So there is much more conformational freedom there. And this affects the calculations as well. So the disulfide data set performs slightly worse than the cyclic peptide one. You can also do this analysis as function of peptide length. And you see that if we are dealing with short peptides, say less than 10 amino acids, here the performance is much better. You see a lot of dark green. So we do generate very good conformations. We are not always able to pick them out. If you look here, for example, in the top 50, we have a high quality model, but our scoring function doesn't pick it out because here we lose all the high quality ones. So, okay, scoring is not a solved problem, even with AI. The longer one, not surprisingly, are the ones that are much more difficult to model. So we do generate reasonable solution, but no higher quality one. And that's pretty much the state of the art these days. So we have this cyclization protocol. So you start from a sequence, build it in extended conformation, then cyclize and sample conformation. And the secret here is to turn off electrostatic to get more bond-like conformation. And we perform an ensemble docking protocol where we give multiple conformation at starting points. Of course, in this example, we assume we know the binding site on the protein. Okay, so if you don't know the binding site and you're going to do a blind docking, that's going to be way more difficult. So this was the peptide story. Glycans very shortly. Same story. So the peptide story was done originally with the HADDOCK 2 server. I only showed you that we can do it now in HADDOCK 3 as well. So we have the cyclization protocol. The glycan story was done purely with HADDOCK 3, the new version. And here we are doing something which we could not do before, as I'm going to explain. So what we have here is a set now of about 90 complexes. So these are protein binding to a glycan. So these are not glycosylated protein. No, it's a protein recognizing a glycan unit. And we have different types of glycans. So some are linear, some are branched. So glycan can be branched, so that makes the conformations more complicated. Some are very short, 3 or fewer monosaccharide units. So shorter means less conformational freedom, easier to model. Longer is going to mean usually trouble. And we generate the glycan conformation using the glycan server, where you can basically build your glycans and generate a 3D model of it. And this is the quality of the predictions from glycan. So you see here RMSD from the true structure, the bond form. The short one, the median, is at 0.9 angstrom, so it's doing a very good job. The long and linear one, you see that it's much more difficult to have 1.6 median, and the long and branch one, same story, with some outliers up to 3-4 angstrom. So if you are at 3-4 angstrom, it's not very likely that you're going to get a good model at the end of your workflow. Okay, these are still large conformational changes. Another protocol is something that you could not do on the classical version of HADDOCK, because now we do a rigid body docking. And usually what we are doing before is select the top 200 model and then refine them. And now we say, no, we're not going to do this selection, but we're going to do a clustering of all the solutions that we have from the rigid body docking. So 5 or 10,000 solutions. And then we select models from all the clusters, irrespective of where they are ranking in scoring. And then we refine them. So in that way, we hope to rescue basically models that might have not made it through the classical scoring, will be still taken to refinement, and refinement, the scoring might change a lot during refinement. And these are the results. So these are unbound results. So again, we start from probably AlphaFold models of the receptors, the glycan server model of the glycans, the oligosaccharides, and you see here what's happening. So this is the success rate, same story, but now in terms of RMSD in this case, high quality will be below 1 angstrom, medium below 2 angstrom, and acceptable below 3 angstrom. So at the rigid body, we sample, not sure if it was 5,000 in this case, we only show the plot up to 200. So you see that in the top 200 model, this is what HADDOCK 2 series will be selecting further for refinement. We only have about 50% of the case or less that have something which is reasonable. If we do a clustering approach, which is possible now in HADDOCK 3, we see that this is a big difference, because now we are selecting 50 clusters, and we ask HADDOCK, give me 50 clusters out of the model that I generated. And you see that the success rate of those clusters now goes much higher. So there are models, so for more than 70% of the case, we have something reasonable, which we then take through the refinement process. Does flexibility help? We are doing this flexible refinement by molecular dynamics. Does it help? You see the result here. So these are 3 examples. So the refinement improves both the conformation, so it means we get lower RMSD, and also the scoring. So this is a short peptide. So in magenta colors, you see the conformation after rigid body docking. In orange, the flexible refinement, and in gray will be the reference structure, the crystal structure of the complex. For small one, you see it's already doing quite well at the rigid body stage. For larger ones, so we have a longer linear, you see that the rigid body model doesn't resemble at all the correct solution, but after refinement, it's getting much closer. Okay, at the rigid body, we are about 4 angstrom. After refinement, we are down to 2 angstrom, and it's ranked number 1 in our scoring function. At the rigid body, it was number 60. This is a branched glycans, which are even more complicated. So you see the best model that we get after rigid body is 5.5 angstrom, but it ranks at position 740. You will never look at that one. After refinement, it ranked on the top, and it has an RMSD of 4.2. It's not great, but that's the best model that we generated, and now you can identify it as one of the top. So we have basically this new protocol based on HADDOCK 3, where we introduce a clustering step, excuse me, after rigid body. And this improves the performance from 33 to 50% of acceptable model being selected for further down refinement. The refinement does improve both ranking and quality of the model. And this is true not only for glycans, but for all different types of complexes. But of course, there's still room for improvements. These remain difficult molecule to model because of the complexity, especially if you have long glycans and branched one. So it's not the end of the story, basically. Now to wrap up, I'm very happy to stand here in front of you after more than 20 years that we started developing the software. You know, it has been a hard effort of a lot of people over the years. We still see an increase in our user base and usage, and that's it has been a lot of work over the years. And even in this AI era now, where we see everything being taken over by AI, there is clearly still a need for physics-based modeling, which is good news. And I introduced you, and now you're going to get your hands on it after the coffee break, to the new version of HADDOCK 3, which is where the future is going. So we don't do much development anymore on the previous version. We fix bugs if we encounter those, but most of the development is in the new version. And there are things in the new version that are not yet available that were in the old version. So this is a work in progress. So this is the current team. Maybe you can guess on which date the picture was taken. Funding from national project and a lot of European projects, with BioExcel Center of Excellence playing a very important role here, because that's one of the few funding schemes which support software development. Many of a funding scheme, you have to solve a scientific problem and you hide a bit of software on the side. But BioExcel is really instrumental for software development, so very important for us, but also for the other teams and partners in BioExcel, and you will hear from the other speaker about that. The team over the years, we came together 2 years ago to celebrate the 20th anniversary. The first publication of HADDOCK was in JAX in 2003. So we celebrated this 2 years ago with a fun meeting, bit of science, a lot of fun. And with that, I'm finished, and thank you for your attention.
So again, online, you can turn on your microphone or type. Better to turn on your mic. And in from board.
I'm very new at protein docking, but I studied the blocking the dimerization by using a molecule. And how can I use the HADDOCK with the seeing the interactions broken in the protein-protein interaction dimerization changes?
Okay. Yeah, so first, so you want to block, so the question is maybe for the people online, so I want to block dimerization of a protein by devising some small molecule that will go in there, or a peptide or something. So the thing is, so if you were to give your small molecule and 2 proteins and dock it in HADDOCK, you will always get a complex. Because by definition, when we do docking, we assume that the molecules are interacting. So what the software is trying to do always is to put the molecule together in one way or another. So you will never get a solution where one protein stays apart and the other one binds. Okay, so that's, you cannot really do that, basically. So in HADDOCK, you could only try to test if your small molecule gives you a nice binding to the interface, but not much than that. The other thing is when you know, these kind of things have to do with binding affinity. Binding affinity is related to free energy. And when we do scoring in HADDOCK, or when you do even physics-based calculation, the energetic that we are doing, it's not free energy. It's energy at best, maybe enthalpy at best if you're lucky. The HADDOCK score that we are reporting, they don't have units. We don't want people to think this is a free energy of binding. It is not. So that's a very hard problem, basically. So no good solution.
Injection to his question. For example, you use HADDOCK to calculate the docking score of these 2 molecules. And it is proposed molecule, for example. Then we calculate docking score with, let's say, this molecule is binding to one of them.
So you want basically to compare the score of the complex to the score of the protein plus small molecule. this is again difficult because scores are not binding free energies. And that's, so, if you compare the score of one protein with another one when you start doing mutations, maybe you can do that. But I would think probably the binding interface in your protein-protein complex is going to be much larger than your small molecule protein interface in this case. And meaning that probably the score that HADDOCK will give you for the protein-protein complex will always be better than small molecule. That's my guess here. So I think, you can compare scores when one component is very similar or the same, and then you make small modifications. But if you take two different complexes, the score might be completely unrelated, and one might be much stronger binding affinity, but a worse score than the other. So there is no correlation between scores and binding affinities if you compare different complexes.
Just same complex, but just addition.
Yeah, so they're same complex.
Component.
So no, not benchmarks. So it's a hard problem.
I mean, you still see in papers people reporting these.
Yeah. But there are some software that are reporting. If you do small molecule docking and you use AutoDock gives you a delta G as a score. And people do papers where they purely do computational working and say, well, I get a delta G of -14, hence it's a good binder.
Okay, but may I have a question? So how big systems, in terms of the amino acid bank, can you handle in reasonable times? Like if I were to dock into a Cas9 thousands.
Yeah, so it depends a bit on, So if you do a two-component system, so if you do multiple proteins, this increases the complexity. What we see on the server is that people now these days do quite some large system where you have several thousand amino acids and it still runs reasonably fast. So there are different factors that define how fast it's going to run. So the size of the system is once because we do energy calculation like molecular dynamics, or your scaling will be at best n log n. And the other thing is how you define the interaction restraints. If you say I'm defining one restraint to the entire surface, that's going to be a lot of distances that you have to calculate, and then you get to an n-square problem in terms of restraints. So that's one bad thing. But we see, and the other limitation that we have is that we're using the old PDB format still, which is limited to 999 residues. Okay, but we see people doing sending the ribosome to the server, or small ribosome units. So we see system of several thousands. On the server currently, we also have an implementation of the Martini force field. So you can coarse grain your particles. So this will limit by a factor of 4 about the number of atoms, and that speeds up the calculation, of course. It smooths also the energy landscape because you don't have all the atomistic details at the interface. And then the last stage will be to return to a full atomistic model. We are currently putting that into HADDOCK 3, and actually Raphael has been doing this work, and we are close to merging that branch. So it's not going to be part of the first, so we don't have yet a first production release of HADDOCK 3. It's going to come very soon. We are finalizing the paper on that. And after that is done, we will probably, the next version will have the Martini incorporation. So then to go to larger system, then it becomes faster.
So another small question. Can you handle systems with large post-translational modifications?
Well, large
protein attached from the center to another protein and
yeah, that's very hard. So what we have been doing is polyubiquitination, for example. So we've done these kind of things. So you can, we had been doing work on that. You could do it. So it's an isopeptide bond. So in principle, you could, it's hard to detect automatically, but in principle, you could tweak the codes to create the bonds, or you could use a distance restraints to put it there. But it means that the bond will never, if you play with distance restraints, we've also been doing what is called covalent docking sometimes. But if you don't have the explicit bond defined, it means that the van der Waals radius of the atom will prevent that they get at a proper distance for the bond. Okay, so it will always be longer, but you can use distance restraints to impose that, yes. And the standard transformer, phosphorylation, sulfonation, some methylations we have supported. Yeah.
Other questions? A few questions on the chat.
Yeah.
Yeah, so
Can we use HADDOCK tree for docking intrinsically disordered proteins with peptides or peptidomimetics?
You can, but it will be probably, in my opinion, a waste of time. Because intrinsically disordered proteins, by definition, have not a very well-defined conformation. So you're going to generate a lot of spaghetti. And what do you make out of that? I'm not sure that it's worth the computational effort. But in principle, you could do that. But yeah, and peptidomimetics are not supported. So we don't have the force field parameters for those. If you have so that's the other problem. So you will need to support that.
What about binding two small molecules to one protein?
That's possible. So you can dock multiple molecules. So I think we have a limit, which is kind of arbitrary of 20. But you could do fragment-based drug design, for example, where you form multiple small molecules at the same time. You need to have the topology. So in a server, the web server can handle up to 20 molecules. I will not do a competition experiment in that case because, if you say both molecules should bind, both molecules will end up there. But maybe there's a preference for one going binary side to the other. We never tested this kind of competition thing, but it would be an interesting project. In HADDOCK 3, you will have to generate the parameter for those molecules.
So does HADDOCK 3 have any limitations or ranges to do this?
Any limitations on?
Any limitations or ranges?
Ranges, on what? Limitation on what? Limitations.
Yeah, maybe.
But maybe they already got their answer.
Yeah.
And is it possible to work with graphene plus protein?
I know that some people tried that in the past. We don't have topologies for graphene, so you need to build the system. So I'm not sure. I don't know. I've seen people trying on a server, and I'm not sure if they were successful or not. So the server used prodrug to generate automatically topologies, and I have no idea if this will work. Other ways of parameterizing graphene will be to use AC pipe. But again, I don't know. In principle, yes, I know people have been using HADDOCK to dock on surfaces also. The main problem here are the parameters and the description of the system. It's not the methodology itself, but it's the energetics, the parameters that you need.
So as you know, RNA protein correlation is part of RNA current situation. Do you have any advice that we can increase its correlation ability?
So RNA protein correlation. I'm not sure what they mean by that. So RNA is difficult, so yeah. Yep.
Cancerogenic protein, does it consider the pH levels?
Or the pH level. So the way the server does assign. So it's if you have very extreme pH, you could have, for example, protonated aspartate glutamate. So this we have parameter for those, but you will have to decide to define in the PDB file that you give to HADDOCK that this is an aspartate which is protonated, which we call probably ASH, I would think. I'm not sure, but we have a different name for protonated aspartate. So if you know that. The probiotic one are the histidines, because histidine has a pKa of 7.4 or 5. So they might be protonated or not. So the way the server does it is we use mole probability to basically build all the hydrogens and then look at the hydrogenable network. It's the same way that GROMACS does, I think, to assign histidine state. Because histidine could be protonated or neutral. And if it's neutral, there is 2 positions where you can put the proton. In HADDOCK 3, the way we do that is by calculating the electrostatic energy. So we generate, when we create the topology, we generate all 3 possibilities for histidine, and we select the one that gives the best electrostatic energy with the neighbors. If you know your protonation state, you can impose it. Okay, but then you need another method to define what you want to have. Yeah.
Okay, so if other questions arise, you can ask during the coffee break, which is now 20 minutes. So we will meet back here at 12:30. Thank you very much.
So let's get our hands dirty on the keyboard. So we're going to run a tutorial using the Truba resources. The tutorial is in the Slack channel, so you have a direct link here. Okay, to find the instruction. I'm also going to show you where you can find all the tutorials. So if you go to bonvinlab.org, this is the website of our group. There is an education tab. This is where we have a lot of course tutorial information. There are also some pre-recorded lectures. I heard this lecture is being recorded as well and so it will be shared, so maybe we will put there as well. And we have tutorials about HADDOCK 2.4, so this is where you find tutorials, a lot of them using the web server. And today we're going to go to HADDOCK 3 tutorials. There is a user manual that explains a lot of things. This is new, so we put this one online very recently. There is a manual to generate restraints for HADDOCK, because HADDOCK 3 you don't define passive residues like the server if you have used that, but you need to generate the parts before giving those to HADDOCK. And today we're going to do this antibody antigen docking tutorial. You see there's a protein glycan modeling tutorial. So this is based on the glycan story I gave you. And we have a basic protein DNA docking tutorial. If you go into the HADDOCK 2 series tutorials, so here there are a bit more things. So we have a basic protein, antibody antigen, that's the one we are doing here today, but then using the web server in that case. Protein DNA, cross-linking tutorial, some ab initio docking tutorial with symmetrical systems, small molecule docking tutorial, ligand binding site tutorial, where we don't assume that we know where the small molecule binds first, and then we do docking against the entire surface analysis, and then targeted docking in the second stage, and some other ones. So there's a lot of information there, so you can always come back and visit those on your own. So for today, we're going to this one. So we don't need to, so we have 1 hour power, which is not a lot. And there's a lot of things here, so we're going to do the basic one. There's a number of bonus things that maybe we can do one depending on the time. One of them, we do the same complex, but using AlphaFold 2 in this case, or AlphaFold 3. So I already talked about antibodies. We don't need to go over those anymore. By the way, while I'm speaking there, I hope you all have access to the Truba resources. You should have started the session, the terminal session, either straight at SSH or through on demand. So here we assume that we know the binding site on the antibody, and we'll have some information also on the antigen. So all the tutorials that we have online have the same structure. There is a color coding. So if you see something with an orange color, this is a question. You should think about it and try to answer it. A blue color will be an instruction. So we have to do something. Green will be visualization commands. In this particular tutorial, it's Pymo. We're not going to run Pymo from the computer that we are accessing, but I might show you some stuff from my laptop and some visualizations also on the tutorial online. And black will be Linux font. So this is command line terminal where you need to type things. In principle, you can copy paste those comments from the tutorial page into the terminal. Some tutorials are the one that I use in the web server will not have any Linux level, so they can be run without Linux expertise. Now we can skip the HADDOCK stuff because you know that. And we go all the way down to the EuroHPC workshop in Istanbul. So you see here setup, software and data setup. So if you want to run things on your own on another system, you can download the archive with the data that you need to run the tutorial. Don't do that now because the data are already on the system. Okay, so go directly to the EuroCC step. So you need to then start a connection with Truba To use the system, you need to be under the Scratch directory, so you all have your own directory. If you run into problems, by the way, you can just raise your hand and Bella can try to help you. So you need to unpack the data on your Scratch directory. Okay, so you have to. And once you are there, then you can copy those commands. Now you are going to copy a zip file, which is on the system already, which also has some scripts that are specific to the infrastructure that we are using, so that it is a script ready for Slurm submission. So you need to do that. So I've actually, in my own directory, I've already everything there, but I will just repeat it. So I'm on the Scratch. So I'm basically now unzipping the archive. So what I'm doing is those two commands that I copied here in my Scratch directory. And then I go into the directory that was just created, which contains those files. Okay. Then to use HADDOCK, you need, so HADDOCK has been installed in a user which is common to this workshop. So we installed it, it's not installed using Conda. Some of our software sometimes are installed using Conda, but we use VN under Python to define an environment for HADDOCK 3. So if you source this file, this command which is highlighted, this will activate HADDOCK 3. And then you can test if you say HADDOCK 3 - H, you should be seeing this in your terminal session. Okay, so let's test that everything is still. And you see that the prompt has changed. It says HADDOCK 3 here. So now in the HADDOCK 3 environment, you can type HADDOCK 3 - H. I'm getting this prompt. So now we are ready to go. Software is installed. The sound is on there. Do I need to do something to activate this microphone?
I think that's fine.
Yeah, it's fine.
I checked it via Zoom and it was fine.
Okay.
Let me check it again.
Okay, so hopefully it's working. So if you have all unpacked the data, everything is there. We are ready to go. There are, by the way, installation instructions in the tutorial. If you were to run it on your own on your computer, then you can try to run things. So the first thing we need to do, so actually the tutorial, if you just, let's take a look at what is in there. So when we unpack, good, yeah. So when you unpack the archive, this is what came out of it. Actually, I realized that there's already a run that has been pre-calculated. So for the sake of this tutorial, I'm going to delete it so that we can rerun it. I'm also going to remove the Slurm output. Okay. So what do you see here? So we're going to get PDB files from the PDB database and process them for docking. But in principle, everything you need is there already. So these are all the, basically the output of some of the files that we are going to use. You also see AlphaFold 2 models, AlphaFold 3 models. So these are those guys here. You see AB Baldy Builder. It's another AI model. This was the PDB directory. There's a restraints directory. This contains restraints that define basically either the true binding site or the paratope from the antibody. So there are different scenarios. So these are files. The tutorial explains you how to generate those, but they are already pre-generated. You have then the runs. So the runs contains a number of pre-calculated results so that You might not have to wait for the results to analyze those results if you want to look at those, okay? And then there is a script directory. So this contains a few simple shell scripts to extract some statistics from the run so that we can analyze the results. We're going to use those. And the last directory in there is the workflow directory. This contains the HADDOCK workflows that we're going to execute. Okay? So before we start, if you look into your directory, you see that there are two files. There's a run HADDOCK free Barbon and run HADDOCK free HAMC. So these are two of the clusters that we have access to. So if you want to run your first docking run yourself, so let's take a look at what's in there. Don't copy my command. If you don't know VI, don't do VI things because you're going to get into trouble. So this is a script which was shown to you in the first session this morning for submission to the system. So, okay, so we are requesting one node. These nodes have 54 cores, and this is the HAMC system, and we are requesting only 30 minutes. Actually, on HAMC, this particular workflow here is going to take less than 5 minutes to run. Okay, so we should all get your results by the end of this tutorial. You see that they are, so what is it further doing? It's basically activating the HADDOCK free environment. That's the first line here. And then it's calling HADDOCK free with the workflow that we're going to execute, which is the one in the workflow directory. Okay, and this one uses all the data that are provided already. So there are other options here. So these are other scenarios that are part of this tutorial. So if you want to run a different scenario, you should comment the first line and then uncomment the next one, or you could uncomment all the lines in principle and run all the scenario in one go. But then you probably need to increase the time that you request to the system because probably the first 4 scenario will be 5 minutes. The ab initio one will have much more sampling done. So this is probably going to take a bit longer. So I don't know, you should probably put it to 2 hours to be on the safe side. And in principle, you could run everything in one go. But every one of you will use one node in AM3. I'm not sure how many nodes are available in AM3, but some of you might be waiting for others. So I would say leave it as it is. And now we can, before we start with the explanation of the tutorial, let's just submit this because it's going to run. And in the meantime, I'm going to take you through the steps to basically prepare the systems. And then when it's finished, we can analyze the results. So the command to submit the Slurm system is sbatch with the name of the script. And that's it. And then with SQ, you can figure out what's happening to your job. You will probably only see your own job. So now I see my own job that has just been submitted. So it's pending, and now it's running already. Okay. So in 4 minutes' time, it should be finished. So if you have all submitted your jobs, we can go back to the other steps. Okay, so once you have unzipped the archive, go into the directory, type this command, and you are in business. It's still it's under the scratch directory. When you do so basically what we did this part. Okay, so you should go to your scratch directory with your username, unzip the zip archive, which creates a directory called HADDOCK free antibody antigen. Go into that one, and then submit the run HADDOCK free HAMC script. So you have?
I have 5 directory, but nothing else. Should I have more?
Did you do? Did you do these commands, the unzip? Not wget, but unzip. Yeah? Oh. Okay. Oh, you need to. Oh, one thing. If you submit your run, and run one is already existing, then it's not going to rerun it. It's going. HADDOCK is going to stop. Okay. So if you want to run your own run, you should delete the run one directory from your home from the directory in the HADDOCK free one.
Yeah. Yeah. Okay. Yeah.
Okay, but even if you submit it and run one was there, it doesn't matter. Then you have a run one already, and the results are there. So no big deal. So now we're going to prepare the files for the docking. So no, our docking is running, but this was just black magic and everything was prepared. So now we're going to do the different steps to set up a docking run from scratch. Important thing for HADDOCK, one molecule in HADDOCK should contain only one chain. Okay, so if you know have you all seen the inside of a PDB file? Do you know that a PDB file is a simple text file? Okay, some people have never seen the inside of a PDB file and you only seen it in PyMol or Camera, but so antibodies consist of two chains, as I explained, light and heavy chains. And we need to transform that into a single chain for HADDOCK. And we have to make sure that there is no overlap in numbering. Because if the light chain start at 1 and the heavy chain start at 1, you will have two residue number 1 in your file if you make it a single chain, and that's going to give problems. So what are we going to do? We're going to use PDB tools. And PDB tools are a set of Python tools used to manipulate PDB files, which we wrote. And they are installed together with HADDOCK free. So when you install HADDOCK free, they come together with HADDOCK free. So what we're going to do is to get the PDB files from the PDB database. We use the PDB fetch command for that. We're going to do some cleaning. We're going to select a given chain, the light or the heavy chain. We're going to delete all heteroatoms. In this case, there is no cofactor that we want to keep. And if you look at crystal structure, there will be water there. There might be co-crystallization molecule. So there might be a lot of junk, basically. And you don't want to include the junk in your docking. So you should at least look at the structure and remove what you don't want. Another problem with antibodies, it comes from the way they are numbered. In these hypervariable loops, there might be insertion in the numbering. So you might find residue 100. And then if you look at the PDB, you have 100A, 100B, 100C. And many software cannot handle this A, B, C. So if you keep it like that, you will get residue 100 and the other next 3 residue will be ignored. And then the 101 will be the next in sequence. But then you are losing information. So that's specific to antibodies and possibly nanobodies. So, sorry, that's this step here, fixed number of insertions. So we have a tool in PDB tools that's going to remove those insertions, basically change the numbering and shift everything downstream from the insertion so that the numbering is consecutive. Another problem with PDB file, which might be occurring if you have a high resolution structure, is that you might see 2 conformations of a side chain in a crystal. And they are both present in the file. Now, software like HADDOCK or GROMACS, they will have a trouble with that because you don't know what to do. You have 2 copies of the same side chain. What is that? It's not a normal amino acid. So PDB cell alt lock basically select the location, one of the 2, and it's using either the first location or the one with the highest occupancy. So we're going to keep then only the coordinate of the file. We're going to select a specific region of the antibody. We don't need to model the entire Y-shaped antibody. We know where the binding site is. So if we can make our system smaller, it's going to run faster, okay? So all of this basically is happening in this one command here. So you can pipe the output of one command into the next one. So the first one fetches PDB-N3 4G6K. That's the crystal structure of the free form of the antibody. It cleans it. It selects the heavy chain. It removes the heteroatom. It removes the double occupancies of side chain. It fixes possible insertion. Keep only the coordinates. Select the domain that we want. Clean it. And write it to a new file called _h. So I can copy this. I go into my terminal. And if I do a listing now, this file has been generated. Okay. And this is my PDB file, which in principle only has 120 residues in it. So that's the heavy chain. Now we need to do the same, but for the light chain. Okay. So these commands are very fast. So we go into the protein database and extract things and manipulate. So now we have the heavy chain and light chain, but this has to become one molecule for HADDOCK. So we're going then to use these commands to merge the two molecules into one PDB file, renumber the file from one so that we have a sequential numbering and no overlap in the residue numbering. And we're going to give it chain A. Okay, so it will have a single chain, which we called A.
Yes.
If you want, you can delete the previous one and resubmit your own, but it's going to. You can submit your own if you want. Yeah. Did you submit a run already? Okay, so that's okay, but you don't need to wait for the output because this is the preparation of the run. Okay, we submitted the run so that the run runs, but now we are taking all the steps to go to the submission. So we worked in a reverse order, basically. Yep.
So in the analyzing step, we have to do we have to reindex the initial states of the chains, chain A, B, C, or we don't need to reindex the file?
Once more, so in the analysis?
In chains, we have to do in one chain
yeah,
A.
Yeah.
But in analyzing state, after the process.
So after the process, at the end of the docking, you will have a chain A, which is the antibody, and a chain B, which is the antigen in that case.
We don't need to reindex the file in its initial state, like chain ABC.
No. Unless you want to go back to the original one, then you could revert the changes. Oh. You could use PDB tools also to do that then. Although to put back the insertion, I think we don't have a tool to do that because that's a different thing. But for analyzing the docking results, you don't need to do that. Okay. So our PDB file is ready. So now we need to do the same, but for the antigen. So the antigen, by the way, I didn't explain at the beginning, but the antibody that we are docking, it's Gevokizumab. It's a therapeutic. It's a drug, basically, and it's targeting interleukin-1B. Okay, so the antigen is interleukin-1B. The antibody is... So now we are going to prepare interleukin-1B for the docking. So I go back to my... preparing the antigen. So I'm going again to get the structure from the PDB database, remove heteroatom, remove double occupancies, assign it chain B, and then my file is ready. Oh. Copy. Now you see you have the, so this one is the clean, this is the antibody, and here you have the clean antigen. So these are the two files. Those files also exist in a PDBs directory. They were already prepared. If everything goes right, there should be no differences between what we just generated and what is in the PDBs directory, and there is indeed no difference. Okay, so they did not change the entry in the PDB, we did not change the software, everything is the same. So our PDB files are ready for docking. Now we need restraints, okay? We need to guide the system to dock to a given site. So the way we define distance restraints for HADDOCK, so HADDOCK is, I didn't tell you that in the lecture, but HADDOCK is using CNS as computational engine. CNS stands for crystallography and NMR system. It's an old Fortran code, actually. So HADDOCK itself is Python code, but the computations are done using CNS, which is a Fortran code. When you do a pip install HADDOCK, it comes with a CNS executable, but in some system, you're going to run into libc libraries that are not present. It depends on the operating system, depends on the compilers. So if that happens, you have to recompile CNS on your own and put it in the right location. If you are lucky, everything goes right when you do pip install and you don't need to worry about it. Yep.
In the first PDB fetch commands,
Yeah.
You selected residues from 1 to 120 and 1 to 107. So
yep.
That was based on what?
Because we know which,
Okay.
yeah. So you have to look at your structure, you know.
Yeah.
So if you know the binding site, if you have a huge structure and you know the binding site is one domain, you want to isolate that domain because that's going to speed up your calculation. Otherwise, you are wasting CPU. Yes. And CPU is precious. And if you waste CPU, also waste CO2. Okay. For sustainable computing, green computing, all those things. Okay. For the antigen, so we get it. And here we don't select a given domain. It's a small protein. So now we need to define restraints. So I was explaining CNS, and CNS is derived from Explore, and Explore is derived from Charm. Or maybe you know Charm is an MD software. So that's the historical relationship. And in CNS, we define distance restraints with an assign statement. CNS, by the way, is a scriptable software. So actually, all the docking protocol in HADDOCK 3 are written at a script level. And this is interpreted by CNS. So, and the distance is defined between two selections, so 2 points in principle, but I explain you the concept of ambiguous restraints, so the points could be multiple in a sense. So we have 2 selections between parentheses, and then we have 3 numbers. The distance that we want to target, a lower bound correction, and an upper bound correction. Okay, so the distance range will be distance minus lower bound, that's the lower limit, and distance plus upper bound, that's the upper limit. In HADDOCK, because if you have these highly ambiguous restraints, we use 2 angstrom as upper limit, and 2 angstrom is shorter than the van der Waals radius of the minimum distance between van der Waals radii. But because of the summation that we are doing with sum of 1 over R6, the effective distance that we calculate is always shorter than the shortest distance. So if you were to do the mathematics, 1,000 distance at 5 angstrom, and you put that into the sum, and you take the inverse 6th root of that sum, the number that comes out is 1.67 angstrom. Okay, so this is why we use a short distance of 2 angstrom, because the effective distance is always going to be short. And then we have a syntax for selection, which is, for example, here. So this will be defining a distance restraint between the beta carbon of residue 10 and 200 for a distance range between 10 angstrom and 20 angstrom. This could be, for example, a cross-link distance from mass spectrometry. So we will say, okay, we select seg ID A, which is chain A, residue number 10, and name C beta, that's the name of the atom. And that will be the selection for the second selection is chain B, okay, another molecule, residue 200, and name CB. But who is familiar with PyMol? Any of you using PyMol? Some of you are using, so you might know the selection syntax in PyMol. So actually the selection syntax in PyMol is very similar to the selection syntax of CNS and Explore. And that's the reason, because the person who wrote PyMol, Warren Delano, was a master's student at Yale University when I was doing my postdoc. And it started writing a graphical interface to Explore at the time, which was called AVS Explore. And we were from the command line with Explore/CNS command, we were doing atom selections that were controlling the graphic display. And later on, this became PyMol, okay? So that's why the selection syntax is CNS-like. That's the historical story, okay? Good, and there are many ways of defining a distance range, because here this is between, so 20 minus 10, it's 10 lower limit, and 20 plus 0, it's 20 upper limit. I could write this distance as 10, 0, 10, and it will give the same range. Okay, so there's multiple ways. So now we want to define the binding site on the antibody.
Can I ask a question?
Yeah.
Is there a specific reason why we are giving CB instead of CA? Because CA is more often used.
That's just an example.
We have to specific.
Well, the crosslinks are, if this was for cross-linking distances, the crosslinks are often to the, for example, the side chain or lysine. Okay, so if you want to put it a bit further, the C beta, anyway, the direction of the C beta will always be well defined, the position. It depends on the backbone. You can put it on the C alpha as well, but you will have to add more correction to your distance to account that it's the NH3 group which is linked actually. And then you put your distance on the C alpha, so you have to correct for that. But it's just an example. When we define ambiguous restraints just based on this residue is important, we don't specify atom. So if you don't specify atom, the selection will be all the atom of that residue. Okay, so if you just say residue 10 and chain A, all the atom of residue 10 chain A will be selected. Now, if you have an antibody, you might look at it and select manually the loops, the CDR loops. There are also software that allow you to predict, to identify those automatically. So in this case, we use ProABC at the time, but so we have a list of residues here that we define as being the 6 loops of the antibody. We're not going to do PyMol visualization. So these will be the commands in PyMol. Actually, I can't do it for you, but I will do it from my laptop. So if I go just to show you that it's working. Okay, so this is only one domain of your antibody. And the top part here, let's see. Okay, my window is back. The top part here are where the loops are. So I open it from the command line in this case. So now this command is going to color everything in white, and then it's going to select the binding site, which we call paratope. And this is the list of residue that we are selecting, which correspond to the list which is up there. Okay, so I'm going to copy paste this command in PyMol. Again, if you are in the Truba resource, PyMol will not be there. And in PyMol, I can copy paste in this top window here. I actually selected the wrong sites. And now you see in red, basically, these will be the CDR loops of your antibody. So in HADDOCK terms, we're going to define those residues that ideally these should make a contact with the antigen. And if we do a surface representation, it's nicer. Okay, you see that it's quite a well-defined binding site on the antibody site, and this is where the binding should take place. So that's information that we're going to give to HADDOCK. And basically what I just showed you was also displayed here. So we have a binding site for the antibody, and that's easy. These are the hypervariable loops. For the antigen, we need something as well. In this particular case, there was an NMR study of this antigen where they are basically looking at interleukin 1b, which is labeled with N15, nitrogen 15, and then they titrate the antibody into solution, and by NMR, you can look at which signals are affected by the binding. It's called an NMR titration experiment. And in this table, which is taken from this 2013 paper, the green residues are the one affected by binding. And they have two different antibodies here. But here you say if you titrate this one, this is the one that we are looking at, Jevokizumab. These are the residues affected by binding. So the green one are the one that we're going to select. So we say, okay, this is affected by binding, so the antibody must bind in this region. And this is the list of residues. So we can again, I'm going to visualize those for you. So this is the binding site on the antigen side. Again, in this case, it's quite a well-defined surface. You see that there, for example, here at the bottom, there's a residue which is not affected by the binding, but most likely this is involved in binding. It's in the middle of the binding site. So this looks quite good. It could well be, when you use NMR data, that you see a binding site here and you see something happening here in the back for some reason. This could be an allosteric process. So something binds somewhere and there's a conformational change somewhere else in the system. And this might be in terms of binding sites, this could be false positive information. Okay, and that's the reason why in HADDOCK we randomly delete in principle 50%. But it's, if you have data, visualize the data before doing any modeling to see those things, because you just list the list of residue will not tell you that all of a sudden there's two binding sites, and then what to do in those cases. But again, this is a rather well-defined binding site. But again, you might say, well, this guy here, it's not seen by NMR, but maybe it does make contact. So in HADDOCK concept, we have the active residues, which are these red ones, are the one that HADDOCK wants to kind of force. It's not enforcing because it's an energy term, it can be violated, but HADDOCK wants those to be at the interface. And we're going to define the surface neighbors of those as possible binding sites. Because experimentally, you might not detect always the full binding site. So you want to increase a bit the definition of the binding site. It's better to be too wide in your definition of the binding site than too restricted for HADDOCK purposes. So we need to select the surface neighbors. The server does that for you automatically. With HADDOCK 3, we have commands to do that. Okay, so again, this does generate a nice patch. This is what I just showed you. So we have a HADDOCK 3 command line tool, which allows you basically to define a list of passive residues. So HADDOCK restraints, it's a command line interface. Passive from active is what you're calling from it. It uses 1,4I, 1B clean. You give the list of active residue, that's what we just visualized, and what the command will return to you are the surface neighbors, if everything runs correctly. Yes. It takes a bit more time because you have to calculate solvent surface accessibility, and this will be the list of neighbors of those ones. So this is something that we want to give also to HADDOCK as passive. And you could visualize those on top, and this is what is being selected. And this selection now, this command line interface allows you to do that automatically. The server will do that for you, but now the command line, you can do the same. For the antibody, in principle, in this case, we don't need to do that because the antibody, we know where the binding site is. It's the CDR loops. So now we have all the information that we need. So we can generate a restraints file. And again, there's the same command line interface. HADDOCK 3 restraints, and we're going to call as option active, passive to ambig. So this is generating a file that CNS understands. And as input for each molecule, you give a file that contains 2 lines. The first line corresponds to your active residue, if you have them. You can leave it empty if you don't have any. And the second line corresponds to the passive residue, the one that you can just calculate. So for our antibody, we're going to pass as this file, which has the first line, the paratope, the CDR loops. The second line is empty. For the antigen, interleukin 1B, we have 2 lines. The first one are the NMR identified residue. The second line are the surface neighbors. Okay. So we can do this So the files are already, those files with the lines are in the restraints directory. So in principle, if we give this command, this should work. Go back to my working directory. And this is what has been generated. So that's a CNS formatted file. So you see a residue, so the first parenthesis, so that's an assigned statement. So here we are defining a distance. The first selection is residue number 1 of chain A. And then we open the parenthesis for the second selection. And then there's a long list which contains all the active passive residue of the second molecule. So 72 or 73 or. If we go down in this file, so there's a lot of residues. We close the parentheses for the second selection, and we have our distance range. Okay, and for each active residue, we'll find such a line in the file. Those files are already in the restraints directory, and this is what the script that we submitted to Slum was using. So we do not depend. Oh, here I'm actually running that now in my laptop, so okay. Groups. Okay. My run has completed, by the way. Okay. So this is what I was just showing you in the terminal. Okay. We have generated this file. And the distance range, in this case, it's this between 0 and 2 angstrom that, yep.
So, like, as I understand, we are collecting all restraints into one file.
Yes.
This is MB. But yeah. I couldn't understand like how we initially do the first restraints.
Okay,
like,
so for the...
we have to go to the CNS for doing this.
No. So the restraints were selected based on the table in the paper. This was the NMR data, and for the antibody based on the CDR loops, okay? And then what we used is for the antigen, interleukin 1B, we also want to select the surface neighbors of those restraints. Because I told you we want to have active and passive. We want to increase the binding site. So we use these HADDOCK-free restraints, passive from active, to automatically select all the surface neighbors of the restraints that of the residue that we defined. And this gives us a list of residues, which is basically shown here. These are the green residues in this picture here. Okay, so this is increasing the binding site, basically. I have experimental data that points to the red residue, and I want to increase the binding site by selecting the surface neighbors in this particular case. And then once we have this information, we have these files here that contains this information. So it's a text file that contains a list of residues. And once we have those text files, we can use this to generate the restraints. By the way, you can also generate those restraints through a web interface. So if I go to not this, the web portal of the server, there's this GenTBL tools here, which allows you to generate, but you see, you can say chain A, chain B, and then you list active and passive, but you have to select the passive yourself, okay? And in a HADDOCK-free command line, there's a tool to generate passive residues. Given a list of active, given a 3D structure, it will select for you the passive automatically. And you can also change the cutoffs. So there's a parameter that allows you to change the cutoffs. So if I go back, probably, let's see if this works. -H. Optional arguments. So this matches first model, active 1, active 2. I don't think we have -H. So these are all the lists. So actually this one has only one. You cannot change the cutoff at which we define. We have another tool that can do that. So this is defining your restraints, okay? So this you have to do yourself, basically, based on the knowledge that you have. If you write your own restraints file, you can validate it to see that the syntax is correct, okay? That's the only, it's a validation of syntax. Say you forget to close a parenthesis because you wrote it manually, that will screw up completely the calculations, because when CNS interprets those files, you open a parenthesis, it expects that it's being closed. So this validation command line interface will do this validation for you. So I need to generate first the file. I was doing it on my laptop, so let's do it again. So now it's not saying any error. So if let's make an error. So let's say I delete the parenthesis here just to see what it's going to do. You see, you get now an error, malformed assigned statement, line 1. So that means it tells you there's an error in your formatting and then you have to correct it. It's still this weird, all this Python stuff here that comes, but basically here you get information what's wrong, and then you would have to go and edit it yourself. Okay. Another thing to realize also, which is specific for antibodies, is now we are dealing with a multi-chain protein, okay? An antibody has 2 chains, and in principle, we have the 2 domains, and those chains are not covalently attached to each other, okay? It's a non-covalent interaction. For the rigid body docking stage of HADDOCK, it doesn't matter because they are moving as one entity. But when we start doing molecular dynamics, we calculate the energy of the entire system, and those chains, in principle, could start moving from each other. And the protocol that we are doing is a simulated annealing, meaning we start at 2000 Kelvin, and then we cool down the system to optimize the interface. So at those temperatures, bad things could happen. Luckily, most of the structure is frozen, so it doesn't matter. But if you have a non-covalent interface like in an antibody, those could move apart slightly. So to avoid that, we are basically defining a few distance restraints between the 2 chains. And there is a command line interface that allows you to do that. This is this restraint bodies option. So it's going to analyze the PDB and detect if there is 2 bodies that are not connected. Sometimes you have 2 bodies because you are missing a loop in a crystal structure which you did not rebuild. And all of a sudden, your domain are disconnected and they could move apart. So it's not that sometimes it's a single chain protein, but because of what you have and what you don't have model, effectively you have a multi-body system. So this will allow you to do that. And you see that it has detected, indeed, that it's a 2-body system in this case, and it picks randomly 2 distances, now between C alpha carbon in that case. It measures exactly what the distance is, and the error limits are 0. I just want to keep the molecule together by defining 2 distances. Now, if I do the same, but for interleukin 1b, I never tried that before, but let's try. It doesn't give you anything because this is a globular protein which consists of 1 chain. Okay. So now we have all the data. Our PDB files are ready, the restraints are ready. So now we need to define a workflow. And we are going to do basically a classic workflow, which consists of rigid body docking, semi-flexible refinement, final energy minimization, followed by clustering. Since we have a reference structure, which we know, so we know the answer in this case, so we can put this Capri eval, and in Capri eval, we give a target structure so that we directly can measure the quality of the model with respect to the known answer. You can only do that when you know the answer. If you put Capri eval with no argument given to it in a workflow, it's going to use your first model as reference so that you can see where are all the models with respect to the best model generated. And this allows you to generate plot and visualize your talking run, basically. Okay. And as last module, we add a contact map. So this is going to analyze for each cluster in a system, it's going to analyze the contact between the chains and visualize those. So you get a list of contacts as a text file, TSV file, tab separated file, but also a visualization of the contact, which allows you to look at those. And this is the recipe that we're going to write. So we have, usually you're not going to write that from scratch. Well, you could do that, but HADDOCK comes with an examples directory where there are different examples for nucleic acid, protein, antibodies, peptides. So you usually will take one of these workflow or one of the workflow from the tutorial and adapt it to your needs. The HADDOCK web app has an interactive workflow generator and editor. So you can drag and drop modules, build your workflow. So this is going to, so actually it's on the website already and it's usable already. So then you can drag and drop and edit parameters and things like that. So, so this is the workflow and this is how the workflow looks like. So this is, I can show you where it is on disk also because it's part of the data that you downloaded. So we give a name to the run directory. So that's run 1. So if you have submitted this workflow and run 1 was existing, HADDOCK will exist telling you, well, run 1 already exists. I'm not going to overwrite it. The compute mode in this case, we call it local because what we are doing, we are sending the full HADDOCK workflow to Slurm, asking a full node, and HADDOCK will run locally on that node. Okay, they are over execution mode. So we have a batch execution mode, which means that you will start HADDOCK free on the login node, and then each compute unit will be sent to the batch. So you will send a lot of Slurm jobs. Usually the system administrator of your HPC center will not be happy with that mode. So you better send the full workflow, and we give it 50 cores. Actually, the node has 54 cores, I think, that we are using, HAMC, so you could say 54. But based on, it's not going to make much difference because our sampling is very limited. We have a very targeted docking, so we calculate 100 model. If you run it with 54 cores or 50 cores, at the end of the day, the time will be the same because you need to do 2 batch of 50 or 1 batch of 54, and the next batch will be 46. Or you should adapt your sampling to the number of cores that you have on your node. Okay, so if you want to say, I want to use all the CPUs of the node, and the node has 54 CPUs, then I could put a sampling of 108, for example. Then I use 100% of the capacity of the node to model things. So that could be a choice. If you use not HAMC, but the other one, Bonvin, I think, there are 2 systems. Bonvin has 40 cores, I think, or something like that. So then 40 core, you will need to do 3 batches. The first 2 batches will be 40, and the last batch will be 20. So this is the workflow. So we say, okay, number of cores, self-contained, it's an option to avoid problems with long file path on some system. Should not be an issue here, but it's more in the way the machinery works. These are parameters that by default are actually true, so we would not need to define them, but it's doing a post-processing, which is going to do some analysis by default, and cleaning, meaning it's zipping gzipping the files to minimize the size of the system. We define the molecule that we want to dock. So this is 2 files that we prepared. If you were to dock an ensemble of conformations, say you have your antibody, but you have multiple structure of the multiple conformation of the loop, you could give it here a PDB file that contains multiple conformations. And these will be formatted as model and model. You might have seen in the PDB NMR structures are defined like that. PDB tools as a tool to make such ensembles. It's called PDB make ensemble, MK ensemble. So if you have, sample conformation using MD, you want to take 100 snapshots from your MD, you could generate such an ensemble. But realize that the more conformation you put in your ensemble, the more you will need to sample if you want each conformation to be used several times. Because if you generate 100 model and you say I give 100 conformation, it means that each model is only used once, which is not a lot. Okay. And here comes the workflow. So we first build the topology, don't define any parameters. If you wanted to fix histidine protonation state, you will have to do it here. There are examples giving that. Then we do the rigid body. We define the ambiguous file name, which is the one we generated. Actually, it's part of the restraints file. We define an unambiguous file name. These are the connectivity restraints to keep the domain together. Anything that you put into the unambig file will always be used. Things that you put into the ambig category, by default, 50% is randomly removed. It's an option that you can remove if you don't want to use it. There's a parameter to turn it off. But that's the distinction. So if you have data that you trust really, you could put them in an unambig file, then they're always used. And the data that you trust less, you leave them in an ambig file. So that's, you can choose as you wish. It's not because it's called unambig that it has to be unambig. Okay, so it's just a name. The default sampling will be 1000 in principle if you don't change anything, but here we do a targeted docking. So this was a scenario which was actually aiming at doing antibody design with a company where they want to be very quickly be able to generate models of antibody antigen complexes. So we have rather good data, and also for the purpose of the workshop, we just sample 100 models. We calculate the quality of those models with respect to the reference that we know. We select the best 40. Actually, we could select 50 in this case, because if we're using HAMS3, it will be, we have enough cores, but okay. Tolerance is a default parameters, would not be needed, but we accept that some could fail. So it could be that the model explodes because there are clashes or whatever. So 5% failure rate is acceptable in this case. Again, we define our restraints, assessment of quality. We do the final minimization, assessment of quality, clustering based on fraction of contacts. So the contacts that are similar at the interface, selection of the best 4 models per cluster, quality assessment, contact map. So that's the workflow, okay? And that's the workflow. If you want to see all the possible parameters in a module, then you will have to use this command, and then some modules have a lot of parameters. But then you will see all the parameters with a short description of what they are doing. Okay, running HADDOCK 3. So we had these 2 options. I think we use HAMS3 once, so I explain you what this is doing. I was giving last month a workshop in Singapore, and we used a supercomputer in Japan to that, so that's why the instructions here are. And they are different, so you can run it on your laptop. You know, if you don't use Slurm, but you can run it on your laptop. If you have 10 cores, this will run fine. Yeah. Well, you don't need to adapt 10 cores because it will never take more than the maximum number of cores available on the system. Okay, so if your laptop has 10 cores and you define here 50, it will only use 10 at a time, not to increase the load unnecessarily. So this is the mode that we are using here, basically, but with slightly different instructions for Slurm that are adapted to this cluster. And this is a Conda example. Activation, Conda installation. We are using here actually a VN installation. There is a MPI mode where you now use multiple modes, but you need to install with MPI for Pi, and you need to have the right libraries of the system. And so I'm not sure if we can run MPI runs on these resources. I tried, but I didn't get it to work. But in principle, if you could say, you know, I need to do a lot of sampling, I want to be fast, I can use 4 modes at a time, then it means you could generate 200 models on 4 nodes directly. And then you need multi-processing communication, MPI communication. Okay, so now let's look at what has been created. So if I go back, so my run has finished. But we have also the runs directory. So if your run for some reason did not finish, we can go look into the runs directory. So I'm going to look at run 1. I'm going to see what is in run 1. So this is the content of the directory. You see that each module in a workflow has been numbered in the sequential order that you give them to the workflow. Okay, so we started with the topology 0, 0, and then we have rigid body, and the last one is 11 contact map. Then I have an analysis directory. I have the data directory. This contains the input data that you provided. So if I look into data, for each module, they are the data that are specific to that module. So if I look data 00 topo A, I find the 2 PDB files that I gave to the, so in principle, the data that you gave are in the run so that it's an archive of what you're doing. So if you mess up your data outside the run, you can still go back to a previous run and restore what you have. If I look at data 04 flex web refinement, here I find only the restraints because this is what I was defining in a workflow. Now each, if I look at rigid body, for example, you find here we generated 100 models, so they are called rigid body and they are numbered from 1 to 100, and they have been zipped because by default because we have this clean option just to limit space. This is the configuration file for the which is used for the rigid body. So this one, these are all the parameters, okay, that are related to that module, but this is what is being used in the computation. So it's not what you define, but they are default parameters, so they are there, just that you have a reference. So anything that you have changed here should be, like if I grab for the sampling, we set it to 100. So it should be 100 here, okay. And then you see an IO file, that's a JSON file, and it contains a lot of statistics and also the scoring properties and things like that. So, and this IO file is passed to the next stage. So we use those IO files basically to communicate between the modules, but that's very technical. So for each module, so there is these things. For example, I'm going to go look at the cluster FCC module. So you see here clustering output. For example, let's see if this works. Yes, so this is the output file of the clustering module. It generated 4 clusters in that case, and you see which models belong to which cluster. And that's information which is used downstream. There is even an HTML file which allows you to visualize actually that clustering matrix. If I move to my laptop, run 108 cluster, open FCC matrix. Let's see what it looks like. Okay, so you see a visualization of the clustering matrix with the FCC value. So you can visualize the cluster that you have. And if you realize that maybe your clustering is not very good, you could change the parameter. So there's a default parameter for the clustering to define what is similar, but at least this allows you to visualize things and what is what. So that's quite nice. Let me find my, okay.
Are these clusters given numbers in scores, based on the score?
No, so based on similarity only. Similarity. Yeah. And then when you put the Capri, when you do the select top cluster module, it will select the cluster, and then the Capri eval will also, they will be ranked on scores at the end. Okay. And then we calculate the way we calculate the scores per cluster is to select the best 4 models of each cluster. So we want to calculate the score on the same number of models, because some cluster might be large, some might be small. Electrostatic is very noisy. So if you take the average of a full cluster, you might have very good model, but you might have some that have bad electrostatic, and then the average will be shifted up. So we say to calculate the score, we want the same number of models in each cluster that we use for scoring. It doesn't mean that there are no more cluster, but and it's nice if your largest cluster is also the best scoring one, but it doesn't have to be. Sometimes you have a smaller cluster, which is much better, and it probably tells you something about how difficult is it to find the minimum of the problem, basically. Okay. So, and then there's this analysis directory, which contains a lot of data. Now, if you want to extract information about the scores, the best is to look at the Capri eval directory, because this contains a lot of information. And this is a simple text file. We can also visualize everything. I'm going to show you that as well. But if I go into the So this is not this one. This is the selection of clusters. So I go to, for example, Capri-Eval here. So you see here, by the way, Capri-Eval also does an alignment of the chain because we changed the numbering of the FC domain of the antibody. So it doesn't match the numbering. It might not have to match the numbering of the reference. So we do a sequence alignment internally to try to match things. And you see two files here. So there is these tab separated files. So one is Capri CLT for cluster, and one is Capri SS for single structure. So in the SS file, you get the singles model scores. In the cluster file, you get the cluster average score. Okay, so if I look, I'm doing that on my laptop because it's nicer to visualize, maybe. Wow. So this is, well, it's actually not nicer, but so you see here a lot of, so the first line gives you what you are looking at. So you get, so the lines are wrapped in this case, okay? But you see the cluster, this is the model one from cluster one, and where is it located? The rank, by default, this file is sorted according to rank. The score, so this is the HADDOCK score. Then you get the interface RMSD, so it's fitting of the interface on the reference. If we are not given a reference, this number will be 0 because the best model will be taken as reference, okay? So this one means I'm 1 angstrom from the crystal structure of the complex. Then you get the fraction of contact that are correctly reproduced. So in this case, 93% of the contact are correct. Then you get another measure, which is the ligand RMSD. So ligand RMSD, you fit on the first molecule, and you calculate the RMSD on the second molecule. So this gives you larger values. If you fit on the interface and calculate on the interface, you get smaller values. But these are all Capri type statistics. You get a DocQ value, which is a combination of RMSD and FNAT, which is here. And then you get component of the score. It also tells you in this file to which cluster it belongs and what was the rank of the cluster. And then you get component of the score. For example, you find electrostatic, you find the van der Waals, it's alphabetically sorted. The van der Waals interaction is the third bus last. So that will be the van der Waals energy of the system. Okay, and the HADDOCK score is a combination of, this is the electrostatic. So it's 20% of the electrostatic plus 100% of the van der Waals, which is here, plus the restraint energy, which is probably this number here. So there's still a bit of restraint energy. We're not satisfying exactly all the restraints, plus the buried surface, the dissolvation energy, which is probably this thing here. But all the data are there, okay? So you can look at those files. And if you look at the clustering value one, then it looks the same, except that now you also see standard deviations for each value because it's the average over 4 model. Okay, so it has more number per line, but the standard deviations are provided. And then you can start looking, you know, so this is the first cluster ranked by HADDOCK, score minus 43, 43, plus minus 3.5. The second cluster is 107, plus minus 5. And the third cluster in this run is minus 88, plus minus 3. So this is on my laptop, which might not be the same run, actually. This might be old data. So if I go to, what is it? Here we get 4 clusters, I think, in the run that we run here. But you see, again, the first cluster ranked by HADDOCK is significantly better than the other clusters. Okay, so the ranking in this case works quite nicely. You also have to realize that these kind of computations are, when you do molecular dynamics, minimization, these kind of things are chaotic by nature, meaning that if you don't run exactly the same sequence of computation on the same hardware, you might get slight differences. Okay, so don't expect, so if you run the same run on HAMS3 and Barbon, these are different hardware, I would expect small numerical differences. I would not expect that you get a completely different solution, but the numbers will not be the same, simply because of the hardware differences. So I explain you that. So again, what are the statistics, the explanation? And then we have these Capri criteria. This is blind competition and expect to use. So you say, okay, acceptable. High quality is below 1 angstrom, or LRMSD below 1, F0 0 fine, or a DocQ score, which should be below, above 0.8. So if you look at the cluster statistics, we can figure out, again, this does not correspond to what we calculated. So if I go back here, so now I have to find which. So this is the score. This is the interface RMSD, and DocQ is here, for example. So DocQ is 0.82. So according to the Capri criteria, DocQ 0.8, this will be a high quality model ranked on top, which is nice. Okay. Now cluster statistics. So now all of this is kind of cumbersome to look at because you are looking at a lot of numbers. So it's nicer if you can look at some summary. And the good thing is that we have this analysis directory, which contains a report, HTML formatted report, and plots also that you can visualize in your web browser. Don't do that from Truba because then you will need, I'm not sure if there's a browser installed, but I'm going to visualize those for you. So if I go to just to show you how it looks like, I go into the analysis directory. And in the analysis directory, you find such a report for the different stage of the docking. Every time that you put a Capri eval module, you will generate such a report. So we can look at the last one. So there is a lot of individual plots and reporting, but there's a report which contains everything. Okay, so now you see here the analysis of the run that I have on my laptop, which is different from the one. But again, you see the score now. It's a more human readable format. The other one is more machine parsable format. But again, you see the best cluster is here, second and third cluster with standard deviation, with all the terms for the energy terms are there. So this is more readable. Download and view might not work on the web app. Those things should have an interactive viewer. And then you get all these plots that are visualizing your data, basically. So that's nice because what you see here, for example, is the HADDOCK score in this plot versus the interface RMSD. Let's make that a bit larger. So you see the interface RMSD from our target, in this case, the score, and you see that this cluster, which is cluster 1, so these are interactive plots. It gives you the average RMSD of the cluster, and the score is clearly better than all the others. So that's a good scenario. It could be that you get a potato of points, and then you have no clear winner, and then you have a problem. You might have to go back to finding better data. You might not be able to decide. And you see the other plots are the constituent of the score. So you see, for example, dissolvation energy here, van der Waals energy. Our top ranking cluster has not the best van der Waals. The second one has the best. But our top cluster has the best electrostatic. So it wins from the electrostatic, actually, in this case. And these are the restraint energies also. You can also see if you go hover over a point, it will tell you which modalities, what the score is, and what are the value, the coordinate in a plot. So this will be RMSD versus restraint energy. And you can, it's an interactive plot. So if you want to focus on one cluster, you can turn off the others. Okay, by clicking on the cluster. And you can zoom in the plot to focus on, I'm just interested to see what's happening in cluster 1. Okay, so I go into the, where is this option? Need to find it. We should put it more. Let's select this one. I'll probably use the zoom. So that's the interactive thing. So zoom. Okay. And now you see all the plots in this row. So this is working per row, but all the plots have been zoomed on that region. And you can then go more into details what is what. So that's a very useful, and here, so you get HADDOCK score and different metrics. So you get RMSD, DocQ, ligand RMSD, fraction of contacts, or similarity of contacts. And then you get the distribution of scores also as this quartile box plots, basically, that show you things. So it's, again, interactive. So it's an easy way to look at the data, basically. Here, so this is what you see here, okay? So this is what I just explained you. Now, we can look at, how good was the docking and how did things go. So there is this script, which is a simple bash script, which is basically extracting information from this Capri evaluation file and providing some statistics, just for us to understand what's happening a little bit. So you could run that, and it's going to give you some statistics so that we can look at the docking results. So I go now back to the Truba system. I go back into the main directory. And this is, so it's extracting, for each Capri step that we put into the workflow, it's extracting information. Okay, this only makes sense because we know the answer, okay? So this has no value if you work on your own system, but basically it's giving you So 2 was the rigid body stage. So we sampled 100 model, we got 25 model out of 100, which were of acceptable quality. And you see the first model had this RMSD or this DocQ score. This step is after flexible refinement. So you see, we have selected 40 models, okay, out of the 100 for refinement. So 14 out of 40 are acceptable. So it's about 1/3. Here at 1/4. So scoring function is not random. That's good. And you see, for example, after refinement, the DocQ score is improved. So we have improved the contact across the interface, which is what the flexible refinement is doing. And the RMSD has dropped a little bit below 1 angstrom. This is after minimization. So the DocQ has improved a little bit, but the RMSD went up a little bit. And this is after the cluster-based selection, where we select 4 models per cluster. Okay, and you can see the FNAT, and in this case, the ranking is working really nicely.
I'm sorry for
No, sure.
Are they also changing with the batch fit Bonvin and Hamsi as well? Because I am with Hamsi and I got the different results.
Probably they will change a little bit, yeah, but it should not be dramatic.
Yeah, not dramatically, but
Yes. What is more dramatic in terms of change, if you look at what is in a runs directory, so there are all these bonuses that I told you about. So this run one, the run that we started now was started from a crystal structure of the antibody in its free form, okay? If you look in a run directory, and these are some of the bonuses, but we're not going to go over them because we still have 10 minutes before lunch. There is a run which is called run 1 ABB. That's the run starting from the antibody model with immune builder, which is an AI tool. Okay, so that's not a crystal structure, it's immune builder. So we could look at run 1 ABB. Okay, so now I'm using this script just to see, what is the impact of using an AI-generated model instead of a crystal structure. In this case, it's quite nice because actually it's giving models of the similar quality at the end as the crystal structure. What do we have more in there? AlphaFold 2, okay? So that's the run 1 AlphaFold 2. If we do the same here, then you see here, that's not as good anymore. So the best model after the final clustering step at the end of your workflow now is 2.8 angstrom away. And it's ranked number 9. Okay, so the rank is not fantastic. So these are single structure ranking, by the way. This is not a cluster, but a single structure. So AlphaFold, and if you look at the beginning, even at the beginning at the rigid body, the best model was 2.5. So the AlphaFold conformation of the antibody, mainly probably the H3 loop, was not as good as the immune builder one. What do we have more? We have an ensemble here where basically we say, well, let's run immune builder, let's run AlphaFold, and combine all those models for docking. So we generate a more diverse ensemble of starting conformation, and then run. So this is the ensemble one. And you see here again by selecting, so the AlphaFold model was given here. The workflow was slightly different because here we do slightly increased sampling, then we do a clustering step. We select from all clusters something and we refine, and at the end we get something which is even better than what we had from the crystal structure and which is ranking fine. So HADDOCK in some way knew which conformation to select through the pipeline and it scores fine. So these are the different, and there's a run also in there which is ab initio. So this run is basically, well, it's one of the bonus, but just to explain you shortly what we did, we say for the antibody, it's easy. We know the binding site. For the antigen, if you don't know anything, that's a much harder problem. Okay, it's very hard to predict binding site on antigen because there is no sequence conservation on those. These are not biologically relevant binding sites. They are just targeted by antibodies, but they don't have a biological function. So here it's
there is no in silico techniques to the binding site detection for antibody and antigens.
There is no in silico, there is no good tools, no. There are different tools out there. People claim that they can do that well. There's recently, I saw, I think yesterday, a paper from an AI model where they say they are able to do that quite well using ESM2 and another deep neural network. I haven't read it yet, but it's because binding site prediction, one of the most important feature is often sequence conservation. And antigen are not per se conserved. Okay, you don't need to be conserved because there is no functional property associated to the binding site on an antigen, typically. Unless your antibody will target a functional site, then you might detect, but in general, it's very hard to predict epitopes. And that's the ab initio run. So here we sample 1,000 model only, so that's still not a very large sampling. And out of 1,000, there were 7 models acceptable. So here we target, so what we did here for the restraints is the CDRs for the antibody and the entire surface of the antigen. Okay, so we say it should bind somewhere on the surface, basically. And here you see there is a very little number of models, but we do select them in this simple workflow, and they made it to the end even in top position. They are not as good as the other one, but you know, it's 4 out of 16. We require 4 for clustering, so we are lucky. If there was 3 out of 16, they will not cluster and we will not see it. So the single model statistic will be there. So no, if you don't know the epitope, it's still a very hard problem.
Can you also explain the ensemble? When you're in the analysis?
Yes, so the ensemble we just had, so the ensemble was AlphaFold, immune builder, and probably the crystal structure. So that was this one. So here it does work very well as well. So we can look at, let's look at the workflow that we run. So the workflows are all here, okay? So this is the workflow for the ensemble. So now you see that this is defined as it's another PDB that we defined before. This is the ensemble. It contains immune builder, AlphaFold, and I think the crystal structure. The workflow further as rigid body, but now we do a clustering step after rigid body, which we are not doing before. Before we're just selecting. Select the best top model from those clusters, and then we refine those. So it's a slightly different workflow. But here we inserted the clustering after rigid body in this case. And the ab initio one, no, it's again starting from the crystal structure. And the ab initio you see here nowadays, an ab initio mode in HADDOCK is to define a distance restraints between the center of mass of the molecule. And that's only what is used to bring them together. And this is what, this is turning on the ab initio mode, basically. We still use the unambiguous restraints to keep the antibody together, but we don't have restraints specific to the antigen anymore. And in this particular run, we also do this clustering step of a rigid body and then go into the refinement. Okay, so getting toward the end, we can also do some visualization. So the last thing that, so if I go into run one, there is this contact map module that we added. Okay. So here there is also a lot of HTML files that are there, but I'm going to open it from my laptop. Which one is it? No. Contact map, open. Okay, so this is the overall report. So you can look at, there's a plot generated for each cluster. For example, if I click on this one, this is the plot which is generated. So now you see the two chains. Okay, green is chain B. If you go there, blue is chain A. And these are all the residues that are making contact. For example, this is aspartate 56 from chain A. And this one is contacting to lysine 97. So if you hover over the connection, you will find all of that. Okay, if you want to look at it in your laptop, you should download the zip archive on your laptop, and then you can open those HTML files from your browser. So it's color-coded also based on the type of interaction. So this is a simple way of visualizing the contact, basically. And you can look at, so there is one such plot for each cluster. There is also a heat map, so that's a different type of plots. It is this, okay, where you basically have sequence versus sequence, and you see what are the short contact established. So you also see the intramolecular contact in it, but here you see the intermolecular contact. And again, you can go on a region, you can zoom in again, it's an interactive plot, and then it will tell you what kind of contacts are those. So can be useful. You also have a list of contacts, basically, which is, you know, all the list, and it calculates also what is the shortest distance. So these are all the contacts that are generated. It's a very long file. And here you have the interchain contact. So that's the one which we are more interested in. Okay. So this is done per cluster. Okay. And this is irrespective, it's good or bad, it's just based on distance. So it's a distance cutoff. Is there two amino acids closer than, I think we use 5 angstrom for this. Or probably a shortest contact, probably a bit more. No, it reports the C-alpha distance on this column, and what it reports is the shortest contact because you calculate all atomic contacts. So if one atom contact is below a given distance, then it's listed. And it's doing it on a cluster basis. So if within the cluster, it's present. So for example, the one that I've look at these ones, I don't know if you can read it. So this one, the contact probability is 1 here. It means that in all the members of the cluster, this one is present. If you have a very small contact probability, it means it's probably not present in all of them. There is something weird because 0, oh no. So there are two, there is a C-alpha contact probability, and there is the shortest contact probability. So they are different cutoffs, but this is giving you things. So the distance, C-alpha, and the shortest distance between any two atoms, and the probability that this is observed in the entire cluster or not. Again, for analysis, this is providing you basically a lot of information. Okay. So it's almost lunchtime, so the only thing that we can do, or I will do, basically is to visualize the different clusters. Yeah.
Is there a tool to observe side chain contacts as well? Because
so this will, yes, but this contact analysis includes the side chain, of course. Yeah. So in the plot, in the table that was showing, there was a C alpha distance, and the other distance in the last column was the shortest distance between any two atoms. So these are also reported. No heavy atom distance. Yeah. Okay. So if we go into now visualization, lunchtime. So let's load biomolecular star underscore 1.pdb.gz. Let's see if it can handle it. PDBs. For G6M matched. Yes. So I loaded, for the run that I have on laptop, which has only 3 clusters, I loaded them and I loaded the crystal structure of the complex. Okay. Going to just, yellow is still, and now I'm going to align everything onto the crystal structure. So now we can see which one. This was cluster 1, which was the top ranked cluster based on HADDOCK score. And you see that it has done quite a good job. And the other clusters, cluster 2, you see that there is it's quite different. So it's a bit shifted and rotated. And cluster 3, now it's a basically 180-degree rotation almost. With reflect, but they all put the same interface, but it's a different rotation because we define the interface, and of course the interface end up at the end in the right region, but the rotation is different. Okay, so you have run your first HADDOCK 3 jobs, I think all completed, I hope, in a system. So data are there. By the way, there is a log file, so if I go back to the so my run one, so there's a log file generated by HADDOCK, which is telling you, but if you run interactively, this is what you will see appearing on screen. But okay, so there is it says when did it start? It first reads the workflow, define the steps, and then it starts running the topologies. It was using only 2 jobs because there were 2 proteins, and then it starts the rigid body modules in batch of 50. And you can see how much time took the rigid body. So it was very fast. Selected figure, so preparation, and then so it's preparing files, then it's running files. So it started at 25 and it finished at 45:20. So it took less than 1 minute to generate the 100 models. And then you go into the refinement and everything, the minimization, contact maps. So a lot of post-processing actually could be, if you are limited in CPU time, you just want to generate model on the system that you have, and the post-processing you could do on another system just to generate plots. It's not a lot of time. And at the end, it tells you that this run into 4 minutes and 25 seconds on the system, which is quite nice. I think so this is HAMS3. If you run on Bonvin, I think it takes 7 minutes and something, or 8 minutes, because the processors are not as fast, but it's still okay. I think on my laptop, it will run also in 6 or 10 minutes with 10 cores for the same thing. Okay. Yeah, and it depends on your sampling and things, what are the settings, but in principle, and this is a limited sampling, okay? We sample only 100 and we find 40. So if you go to the full workflow, it might be a factor, it will not be a factor 10 more. We sample 10 times more by default, which will be 1,000 at rigid body, but we refine only a few hundred. And the refinement is what takes more space. So probably it will be a factor of 6 to 8 times longer. So it will still complete, I would think, in probably half an hour or a bit more. It should complete on one node.
can we increase out of 10 as well for getting much more clusters?
So you can go, or, you know, it depends on the computing resource that you have at the end. And it depends on the information that you have, because if you have very well-defined information, if you do much more sampling, you will not end up probably with many more clusters, because at the end, you're always targeting the same interfaces, and the electrostatic and the shape will have preferences. So you might get, you know, 6 clusters at the end, but you're not going to get 100 clusters by doing many more sampling. Maybe, yes. Maybe you optimize the scores, yes. Yeah. And sometimes, you know, when you sample a lot, you also get more false positive. So bad models that might score better. So then you get a scoring problem at some point. So that's, it's a complex energy landscape we are moving on. So you might end up in local minima. We're not going to do the bonuses, by the way. So you can look at them. Arctic 3D is a software that extracts information from the PDB and reports on all the available interface that the protein used to interact with other molecules. And it clusters those. So if you are working on a system, you might want to know, is there a structure in a PDB? What is it doing? You will extract a lot of information. It's going to cluster those. It's going to tell you information possibly. Is there a clustering of function also? So you might see that different interface might have different function. So it's all non-knowledge from databases, but it's just giving you this information. And then was about using the AI model. I showed you that, for example, in this case, Immune Builder was giving you better results than AlphaFold. And then there is running AlphaFold. This was the ensemble docking. I just show you something about it. AlphaFold 2. So it's explaining you how to run it, but I would expect that many of you have used it before. What is surprising in this case, this was a structure solved in 2013. So it's part of the training set of AlphaFolds, okay? And if you look at the PAE matrix for this system, if you know what this means, blue means reliable, red means I don't know where to put this domain with respect to the other. So the block that you see here is the antibody-antibody interface. This is the antigen. And basically, those matrices are telling you that AlphaFold is not confident on where to put the antigen with respect to the antibody. And it was part of the training set. Okay, so it has no reliable prediction in this one. The confidence statistics are quite high. Okay, it gives you PLDDT of 90. Again, if you don't know what this means, you can ask me, we can discuss, although the interface statistics are bad. And if you visualize those and look at those with the epitope, which was identified by NMR, there is not even a single conformation generated, but this is only 5 models, okay? Maybe you have to generate 1,000 AlphaFold models, but there is not a single conformation that puts the epitope identified by NMR in the interface. You can run AlphaFold 3 on those. AlphaFold 3 does put some epitope on the right region, but again, there is no correct solution, basically, even AlphaFold 3. So it does, they do claim much better prediction performance on antibodies, and they have a better prediction performance, but they have not solved the problem yet. Okay. And by the way, if you are the last bonus here, it's a scoring scenario. Just to explain. So you have generated models with AlphaFold or with Plus4, with some other models, and you just want to get the HADDOCK score out of it. So you don't need to do any docking. You give an ensemble structure. It's going to run a short minimization and return the HADDOCK score out of it. So that's, for analysis purposes, it can be nice. That's it. Okay.
Well, thank you very much. Just in time, I should say, because we never take 1 hour to eat. We like to work a lot in this place. So, but if you have questions, you can always catch dive lunch in the days. But we meet back at 2 o'clock. Nandan is going to be doing the products at 2 o'clock. So completely new stuff or maybe not. A lot of overlaps. So to go to lunch, you go downstairs, go to the other end of the corridor, and we'll meet you there.
Il n'y avait pas de...